Welcome back to IMAT Tutor. This is the one place where every IMAT subject, every topic, and every past paper trap gets broken down the way the exam actually tests it, not the way a textbook lists it. Today we open one of the highest yield anatomy blocks on the whole exam, the skeletal system and joints. Before we touch a single bone, I want to change how you see this topic, because the way most students see it is exactly why they lose marks on it. Most students look at the skeleton and see a coat rack, a dead frame, something dry you memorized the night before and forget the morning after. And if that is how you see it, the IMAT will punish you, because the IMAT does not test the skeleton as a dead frame. It tests it as a living, breathing, constantly rebuilding organ that is deciding right now inside your own arm how much calcium is in your blood. Think about that for a second. The level of calcium in your bloodstream has to stay inside a razor-thin window, because calcium runs your heartbeat, your nerve signals, your muscle contractions, and your blood clotting. Drift too low and your muscles will cramp and your nerve will misfire. Drift too high and your heart rhythm goes wrong. So the body needs a giant, reliable warehouse of calcium it can draw from in seconds and pay back over hours. And that warehouse is your skeleton. 99% of all the calcium in your body is locked inside your bones. So every time we talk about a bone cell or a hormone or a bone disease in this lecture, we are really talking about how the body manages that warehouse. Hold that idea. It is the spine of everything. Quick thing before we go deep. Everything I'm teaching from is on screen right now and you can have it for free. These are the anchor slides. Every keyword, every trap, every pearl laid out lecture by lecture. Download them, keep them open in a second window and write on them as we go. The link is in the description and in the pinned comment. Grab them now, then come back. Here is the second thing that makes this lecture worth 3 hours of your life. The skeletal system sits at the crossroads of 4 different subjects the IMAT loves: anatomy, cell biology, developmental biology, and endocrinology. A question that looks like it is about a bone cell called an osteoblast is secretly a cell biology question about lysosomes and acid. A question that looks like it is about how a bone is formed is secretly a developmental biology question about cartilage. A question about a hormone called PTH is secretly a physiology question about calcium balance. Examiners know most students study this as separate islands, so they build questions that force you to jump between islands. If you can make those jumps, this topic becomes a source of easy points. If you cannot, it becomes a minefield. My job today is to build the bridges. And the third thing, that is negative marking. On the IMAT, a correct answer earns you plus 1.5. A wrong answer costs you minus 0.4. And a blank is 0. That negative marking changes everything about how you should think. A confident wrong answer is worse than no answer at all. And this topic is full of near identical pairs designed to make you confidently pick the wrong twin, such as osteoblast versus osteoclast, endochondral versus intramembranous, osteoporosis versus osteomalacia. Every one of these pairs is a trap the examiners have planted on purpose. So throughout this lecture, every time we meet one of these pairs, I'm going to stop. Name the wrong belief you probably hold, take it apart, and hand your correct version so cleanly that you can never confuse them again. So let me walk you through the whole read ahead. And as I say each item, watch it on your screen, because the order I say them in is the exact order they appear, and it is the exact order we will teach them. We begin with 6 functions of the skeleton and the great split of the body into its axial and appendicular divisions. The axial part is the central tower of your body, skull and spine and ribcage. The appendicular part is everything that hangs off it, your arms and legs and the girdles that attach them. Get that division clean and the whole family of questions becomes trivial. Then we zoom in on a single long bone and learn its macroscopic anatomy, naming the diaphysis, which is the shaft, the epiphysis, which is the swollen end, the periosteum, the living wrapper on the outside, and the endosteum, the lining on the inside. Every one of those names comes with a job, and the exam hands you the job and asks for the name, or hands you the name and asks for the job. From the naked eye view, we drop to the microscopic, compact versus spongy bone. The two architectures bone is built in, and the beautiful repeating brick of compact bone, the osteon, which you will also see called the Haversian system. Then we crack open the material itself, the bone matrix, and split it into its two halves, the flexible organic osteoid and the hard inorganic hydroxyapatite. Understand those two halves and you will predict every bone disease before I even name it. Then the workers, the four bone cells, the osteoprogenitor, the stem cell, then osteoblast, which is the builder, osteocyte, the caretaker, and the osteoclast, the demolition crew. We will spend real time here because this is where most marks are won and lost. We follow that with the osteoclast resorption mechanism, how that demolition crew actually eats bone, and the elegant control system that switches it on and off, the RANKL/RANKL/OPG axis. Next, how bone is born. There are two completely different ways, and the exam contrasts them constantly. Intramembranous ossification is direct bone from mesenchyme, bone built straight out of raw embryonic tissue with no rehearsal. Endochondral ossification is the long way round, where the body first carves a full-size cartilage model and then replaces it with bone. And this is the one with the famous epiphyseal plate zones that decide how tall you get. Then we covered the bone remodeling cycle, how bone rebuilds itself your whole life, and the fracture repair stages, how a broken bone heals. Then the control room, the hormonal control of bone, and the cast of 6, PTH, which raises blood calcium, calcitonin, which lowers it, calcitriol, the active form of vitamin D, estrogen, the great protector of the female skeleton, growth hormone, which drives height, and glucocorticoids, the stress steroids that quietly weaken bone. Each one gets a stimulus, a target cell, and a direction. And if you know all three for each hormone, no calcium question can trap you. After the hard tissue, we do the soft cartilage in its 3 flavors: hyaline, fibrocartilage, and elastic, each engineered for a different mechanical job. Then we assemble bones into joints, doing the full joint classification into fibrous, cartilaginous, and synovial, and then a deep look at synovial joint structure and the 6 joint types, the freely moving joints that let you walk and grip and turn your head. And we finish where the exam finishes, on disease, bone pathology, where every mechanism we build pays off. We will nail osteoporosis, rickets, hyperparathyroidism, and Paget disease. And you will see that once you understand the normal machinery, the diseases are just the machinery running too fast, too slow, or in the wrong direction. That is the whole journey. Now let us take the first step one last time, because it governs how you should play every question. Plus 1.5 for a correct answer, minus 0.4 for a wrong one, 0 for blank answer. And remember that skeletal questions are frequently multi-step, pertaining to a hormone, to its target cell, to a matrix effect, to a change in serum calcium. Miss one link in that chain and the whole answer flips. So we are going to build every link so solidly that the chain holds under pressure. Let us go. Let us build the skeleton from nothing, the way you would draw it on a blank whiteboard. Start with the simplest possible truth. A skeleton is a rigid framework of bone and cartilage. Two materials, not one, and that will matter later. So notice it now. Mostly hard bone, but with cartilage at the joints and in a few special places. In an adult, that framework adds up to 206 bones. Not a round number, and you do not need to love it, but you do need to be able to rebuild it, and I will show you how in a moment. Now, the single most useful move in this entire topic: split the skeleton in two. The exam splits it, so you need to do it too. The axial skeleton is the central axis of the body, the tower running up the middle, and it contains 80 bones. Think of the word axis: skull on top, spine down the center, rib cage around the chest. That is your axial. Then the appendicular skeleton, and this appendicular skeleton is everything appended to that tower, everything that hangs off it: your arms, your legs, and the girdles that bolt them on here and here. And that contains 126 bones. And here is your rebuild trick: 80 plus 126 is 206. So if the total ever slips your mind, you never memorized it in the first place. You reconstruct it from two halves. That means 80 and 126. So the two definition lines on your slide now carry meaning, not just numbers. Skeleton, rigid framework of bone and cartilage, 206 bones in adult, and two divisions: axial, having 80 bones, and appendicular, 126 bones. Axis in the middle, appendages on the outside. Why does the body pour so much energy into building and maintaining this organ every single day? 6 reasons. And I do not want you to memorize 6 words. I want you to feel each job, because a felt idea sticks and a memorized word evaporates in the exam hall. First is support. The skeleton is the mechanical framework holding the body against gravity. Take the bones out and you are a jellyfish on the floor. The skeleton is what lets you stand up against the constant downward pull of the earth. That is the most obvious job, and it is the one everyone remembers. Second is protection. Hard bone builds armored boxes around the softest, most irreplaceable organs. The cranium protects the brain, a helmet grown from your own body. The thoracic cage here, this cage protects the heart and lungs, a cage of ribs around the engine room. The vertebral column, if I draw straight, it's in the posterior side. This is the vertebral column, and this vertebral column protects the spinal cord. That means spinal cord is inside the vertebral column, and spinal cord is main cable of your nervous system. And the pelvis protects the bladder and the reproductive organs low in the body. Wherever nature puts something precious and squishy, it grew bone around it. Third comes movement, and here's the key insight most students miss: bones do not move themselves. Muscle move bones. So what do bones actually contribute to movement? They act as levers. Bones act as levers for muscle pull. And crucially, the bone shape sets the mechanical advantage of that lever. Think of a longer wrench. Let's say this is a wrench, turning a bolt more easily than a short one. They look like bone, but I try to express that these are wrenches. The longer one would be easily rotating the bolt, right, when you move it in counter-clock or clockwise direction. So the muscle supplies the pulling force, but the length and the shape of the bone decide how much that force gets multiplied at the joint. This is a joint example, and this is the elbow joint, which we're going to talk about later. So we can say muscle is the engine and bone is the gear or gearing. Fourth, and now we arrive at the function the IMAT is obsessed with: mineral homeostasis. This is our warehouse from the intro. 99% of body calcium and 85% of body phosphate are stored in bone. Homeostasis just means keeping something steady. The body keeps blood calcium steady by depositing spare calcium into bone. Let's say this is blood vessel and let's say this is bone. The body can bring calcium from the bone into blood, or it can do the opposite when it is required. So we can say that the body keeps blood calcium steady by depositing spare calcium into bone when there is plenty and withdrawing it when blood runs low. The skeleton is a mineral bank with deposits and withdrawals happening every hour of your life. Almost every hormone question later in this lecture is really a question about this one function. Fifth comes hematopoiesis. That long word just means blood making. Red marrow in spongy bone produces erythrocytes. This is the marrow we are talking about, and this is red marrow. Not only erythrocytes, it will make platelets and most leukocytes. Now break that down in plain terms. Erythrocytes are your red blood cells that carry oxygen. Platelets are the tiny fragments that clot your blood, and leukocytes are your white blood cells that fight infection. All of them are manufactured inside your bones, in the red marrow. So your bones are not just scaffolding, they are a blood factory. Sixth comes triglyceride storage. Triglycerides are just fat. Yellow marrow, which is adipose, that is fatty tissue, sits in the medullary cavity of adult long bones as an energy reserve. So now you have two marrows living in the same skeleton, and your jobs are completely different. By you, I meant you are yellow bone marrow and you are red bone marrow. Your job, the right one, is to produce blood, and the left one is to store fat and burn that difference in, because the very next thing the exam does is try to swap their jobs and catch you. Here is the trap. In the exact words you will meet, yellow marrow is adipose tissue serving as an energy reserve. It does not perform hematopoiesis. It is a fat cupboard, not a factory. Red marrow is the hematopoietic tissue, the actual blood factory, and it persists in the flat bones, the sternum, the ilium of pelvis, the ribs, and the vertebrae, and also in the epiphyses of long bones. And here's a lovely developmental detail the exam likes. In children, red marrow also fills the medullary cavity, the hollow shaft, but as you age, it is progressively replaced by yellow marrow. So let's say this is the bone. Inside the bone, you will have medullary cavity. Children have red marrow in it, and as you age, the red marrow is progressed with or filled with yellow fatty marrow. So you can say a baby is a blood factory almost head to toe, while an adult concentrates the factory into the flat bones and the bone ends. Epiphysis is the bone end that we will discuss later. If a question says yellow marrow makes blood cells, it is wrong on its face, and you should feel that wrongness instantly. And here's the pearl that will echo through the rest of this lecture, so plant it deep now. Bone is the reservoir from which calcium is withdrawn under PTH stimulation, and into which it is deposited under calcitonin and estrogen influence. In plain language, one hormone PTH makes a withdrawal from the bank, pulling calcium out of the bone into the blood vessels or into the blood. Other influences, that is calcitonin and estrogen, encourage deposits, keeping calcium locked safely in bone. That means it does the reverse. Now the punchline, and it is the reason osteoporosis exists. Serum calcium is defended at the expense of skeletal mass. The body cares more about the calcium in your blood than about the strength of your bones, because wrong blood calcium can stop your heart today, while weak bones only break next year. So chronic calcium demand therefore weakens bone quietly over years as the body keeps making withdrawals to protect the blood. Every word of this pearl comes back when we reach diseases. For now, Just hold the image of a bank that will empty its vault to keep the blood correct. Now let us actually fill both columns of that great divisions, because the exam will hand you a single bone and ask which side it belongs to. I will read the table the way you should store it in your head. Axial on the left, appendicular on the right. Start with the axial side, the central tower. The skull contributes 22 bones. That surprises people because you think of the skull as one helmet, but it is actually many plates fused together, plus the small facial bones around here. Next, the vertebral column in the adult contributes 26. Hold that 26. We will justify it in a minute because it is a favorite trap. The thoracic cage here that has been isolated and shown around here, which is your ribs plus the sternum, the breastbone, contributes 25. Or we can say this thoracic cage is 24 ribs in 12 pairs. These are the ribs, and with the ribs, you will add one sternum in the center. The hyoid here, this hyoid bone is a small lonely bone in your throat and it contributes 1. And the auditory ossicles, the 3 tiny hearing bones around here inside each ear contribute 6 or 3 per ear. Add those and you get your total axial of 80. Now the appendicular side, the appendages and their attachment points. The pectoral girdles made of the clavicle and scapula. This is the clavicle, one on here, another one here. And if you rotate the model, then you will see scapula or shoulder blades. One is here, another one is here. So we can say the pectoral girdles are made up of clavicle and scapula. Sometimes clavicle is called collarbone, and the scapula, as we have mentioned, it is called shoulder blade. They together contribute 4, 2 on each side. The upper limbs, your arms all the way to the fingertips, from here to up until here, contribute 60, 30 per arm, because your hands alone are a crowd of small bones. The pelvic girdle, the hip bones, contributes to 1 on each side, one over here, another one here. And the lower limbs that has been cut off here but shown here again contains your legs down to the toes. They contribute 60 or 30 per leg. That gives your total appendicular of 126. Now here's a flag I want you to plant right now. The clavicle or the collarbone sits on the appendicular side because it is part of the shoulder girdle. Remember that, because when we reach how bones form, the clavicle is going to pull a surprising trick, and this is the setup for it. Now, why did I insist on 26 for the adult spine when you may have learned 33 somewhere? You see vertebral column written 26, but some people say that is 33. That is shown in this side. And surprisingly, both the numbers are correct for different ages, and that is exactly the crack the examiners try to pry open. So let us build the spine developmentally from child to adult, and both numbers will make sense at once. A child has 33 individual vertebral bones, and you can count them off from top to bottom. 7 cervical in the neck, 12 thoracic where the ribs attach, 5 lumbar in the lower back, 5 sacral at the base, and 4 coccygeal in the tailbone. 7 plus 12 plus 5 plus 5 plus 4 is 33 separate bones in the young child. Then something happens as you grow up, and that is called fusion. The bottom bones weld together. So the adult has 26 bones after fusion, because 5 sacral fuse into 1 sacrum, a single triangular bone, and the 4 coccygeal fuse into 1 coccyx, a single tailbone. So the neck, chest, and the lower back keep their separate bones. 7 plus 12 plus 5 is 24, and then sacrum and coccyx add 2 more, giving 26. Same spine, 2 valid counts, decided entirely by age. While we are here in the chest, lock in 2 more anatomical facts the exam likes. The sternum has 3 parts, top to bottom. the manubrium, then the body, also called the gladiolus, then the xiphoid process, the small tip at the bottom. And the ribs come in 3 types. And for that, let's look at this part of the diagram. From pair 1 to pair 7, that means the first 7 pairs of the ribs. Let's count 1, 2, 3, 4, 5, 6, 7. We have 2 missing here because the diagram is a bit inaccurate, but you just need to know that first 7 pairs are true ribs because they are joining the sternum directly by their own costal cartilage. Pairs 8 to 10 are false ribs because they are joining indirectly with the sternum. And pairs 11 and 12, this one is 12, this one is 11, they are called false or floating ribs. To be more precise, you gotta call it floating ribs because they don't reach the front at all. It was supposed to reach like this, but it doesn't. That's why they are called floating ribs. So every line in that mechanism block on your slide now has a story. We covered the comparison table correctly with the diagram. Then we have talked about the child count, the fusion event seen in adults, and the extra sternum and rib facts sitting alongside. Each one is the end of a short story. You just watch unfold. And here's the trap spelled out. The number 33 counts individual vertebral bones in the child before sacral and coccygeal fusion. The number 26 counts adult bones after fusion. And this is the important part. IMAT stems will specify whether they want individual bones or vertebral segments. So read the wording before you answer. Do not average the 2 into some meaningless middle number, and do not assume one of them is wrong and the other is right. They're both correct. The wording of the question is what tells you which one they're asking for. Slow down on the phrasing and this trap becomes a free point. Now a beautiful little pearl, the kind of single fact question the IMAT loves because it is either known or not known. The hyoid is the only bone in the body that articulates with no other bone. Articulate means to form a joint with. Every other bone touches at least one neighbor at a joint. The hyoid touches none. Instead, it is suspended by muscles and ligaments in the neck, floating in a sling of soft tissue, superior to, meaning above, the thyroid cartilage of your voice box. It is the anchor point for the muscles of the tongue and throat that you use to swallow and speak. And for classification, the hyoid belongs to the axial skeleton, not the appendicular. Pair it with the flag we planted earlier. The clavicle, by contrast, is appendicular. 2 small bones, 2 clean single fact questions, both of which show up again and again. Now we will discuss the macroscopic anatomy of a long bone. Pick up a femur, your thigh bone, in your imagination, and let us name it from the outside in. Because the IMAT hands you a region and asks its function, or hands you a function and asks its region. A long bone is exactly what it sounds like. A cylindrical shaft it will have, which is called the diaphysis, with 2 expanded ends, here and here, called the epiphysis. A pole with a knob on each end. These epiphyses are compared with poles with knob on each end. And you can think the same for femur, humerus, tibia, and the radius, which are the big bones of the arms and legs. Short bones, like the little cube-shaped bones of the wrist, follow different rules. When we say long bone, we mean this pole with knobs design. That means this is the pole and these have knobs on both sides. Now we walk the regions in the exact order they sit on the bone. The diaphysis is the shaft, the long middle portion. It is a thick, compact bone wall, a dense tube, and that tube shape is deliberate because a hollow tube resists bending far better per gram than a solid rod, which is why bicycle frames and scaffolding are tubes too. Inside the tube is a hollow and the shaft encloses the medullary cavity. This region, this area is medullary cavity. And this medullary cavity or this marrow space. We will come back to it later. At each end sits the epiphysis that we have focused. These are the expanded ends. You will notice that I'm repeating the same words again and again with different types of language so that you can catch all the tricks an examiner can play on the exam day. So, as we were saying, the epiphysis, it is built differently from the shaft. It is actually a thin compact shell over a spongy bone interior. Why spongy bone inside the ends? Because the ends bear compressive joint loads. The pounding forces that travel through a joint and spongy bone is superb at absorbing compression, like the crumple zone of a car. Between the shaft and the end is a transition region called the metaphysis, here or here. The flared transition zone, this metaphysis in a child is the crucial site of the epiphyseal plate in growing bone. The epiphyseal plate deserves its own moment. It is a hyaline cartilage disc and it is the site of longitudinal growth, meaning it is literally where your bones get longer. That means the bone will grow top and bottom, and it is literally where your bones get longer, where your height comes from during childhood. When you finish growing, this cartilage disc turns to bone and leaves a scar called epiphyseal line, you see here. The bony remnant after the plate ossifies at skeletal maturity. So a growth plate on an X-ray means a child still is growing. A growth line means growth is finished. Now cover the joint surfaces. Where one bone meets another, the end is kept with articular cartilage. We can see the surface here. And this is the articular cartilage, the light blue portion. And this is hyaline cartilage. You need to remember 3 properties regarding it that make it special. And exam tests them relentlessly. First one is that they are avascular, meaning it has no blood vessels. It has no perichondrium. That means it does not have any covering membrane, and it is nourished by synovial fluid, fed by the joint fluid washing over it rather than by a blood supply. Those 3 facts together explain why joint cartilage barely heals when it is damaged. No blood supply means no easy delivery of repair cells and materials. Now the 2 living linings, one outside and one inside. On the outside, we have periosteum. Break the word down. Peri means around. Osteo means bone. So it is the wrapper around the bone. It has 2 layers. An outer fibrous layer plus an inner osteogenic layer, which will have osteoprogenitor cells. Let me repeat it. The periosteum has 2 layers. The outer layer is fibrous layer. The inner layer is osteogenic layer, which contains osteoprogenitor cells. So this whole layer is periosteum, and the outer layer, it is made up of fiber. That's why we call it fibrous layer. The inner layer, we call it osteogenic layer, which contains osteoprogenitor cells. And that inner layer that we are talking repeatedly, it is gold because it holds the stem cells of bone and it drives appositional growth and fracture repair. Appositional means the bone will grow both in right and left direction. Or we can say that appositional growth means growth in width, thickness. So the periosteum is how bones get thicker, and it is the source of repair cells when a bone breaks. On the inside, lining the hollow, this is the hollow part. The lining, it is endosteum. Endo means inner, a thin lining of the medullary cavity and trabeculae that also holds osteoprogenitor cells. And the space it lines, the medullary cavity, this cavity we're talking, is the hollow shaft interior, which holds yellow marrow in adults and red marrow in children. Exactly the marrow story from the last slide. So we have covered the definition portion. We now know what long bones are, and the model bones are femur, humerus, tibia, and radius. And the every labeled region on your slide now carries its job. Diaphysis is the tube that resists bending. Epiphysis is the shock absorbing unit or shock absorbing end. Metaphysis and epiphyseal plate are the growth zone. Epiphyseal line is the bony remnant after the plate ossifies at skeletal maturity. Articular cartilage. It is a hyaline cartilage found on joint surfaces. They are avascular, they don't have any perichondrium, and nourished by synovial fluid. Periosteum, it has outer fibrous layer and inner osteogenic layer with osteoprogenitor cells. Endosteum is the thin lining of the medullary cavity that we have covered. And medullary cavity is the hollow shaft interior. And here's the trap. It is tempting to think the periosteum wraps the entire bone like paint on a pole. That means you would consider the periosteum spans across the whole bone surface, but that is not correct. Periosteum covers every bone surface except the articular surface. That means this surface is not covered by periosteum. So at the very ends where the joint is, the periosteum stops and cartilage takes over. And remember that articular cartilage has no perichondrium and no blood supply. One more consequence that the exam loves to test clinically. Loss of periosteum at a fracture site. Let's say fracture is here and you lose periosteum in that place. It will severely impair healing because the osteoprogenitor reservoir is gone. The periosteum is where the repair cells live. So if a bad injury strips it away, the bone struggles to knit back together. Periosteum equals repair potential. No periosteum equals poor healing. That is written here, that periosteum covers every bone surface except the articular surface, which are covered by articular cartilage instead. And as articular cartilage has no perichondrium and no blood supply, joint cartilage heals so poorly. Loss of periosteum at fracture will severely impair healing because periosteum is the reservoir of osteoprogenitor cells, and loss of periosteum means osteoprogenitor cells gone, and that equals impaired healing. And the pearl cleanly separates the two ways a bone can grow, which students constantly blur. Longitudinal growth in length, occurs only at epiphyseal plate, and it ceases when the plate closes at the end of puberty. Once that plate turns to bone, you cannot get taller, full stop. Appositional growth in width or thickness occurs at the periosteum. You see in this image like this. This appositional growth continues throughout your whole life. Now watch how this predicts a disease. This is why excess growth hormone after plate closure causes acromegaly, the thickened hands, feet, and jaw, rather than gigantism. If you flood an adult with growth hormone, the length plates are already closed, so they cannot get taller. Instead, they only see appositional growth. That means the growth in width. So the bones of the hands, feet, and face grow thicker and coarser. Same hormone, but whether the growth plate is open or closed decides whether you get a taller person or a thicker boned one. That single logic answers a whole family of endocrine questions. Now we drop below the naked eye to the microscope. Because bone is not one material, it is built in two different architectures, and telling them apart is worth several marks. The first one is compact bone, also called cortical bone, from cortex meaning outer shell. It is dense, solid looking, and makes up about 80% of skeletal mass. Its repeating building block, its structural unit, is osteon. The second architecture is spongy bone, also called cancellous or trabecular bone. It is a porous lattice, an open honeycomb, and its structural unit is trabecula, a little strut. So hold the contrast at the center of this whole slide. Compact bone is made up of osteons, spongy bone is made up of trabecula. So that would be osteon versus trabecula. Everything else hangs off that. Let us build one osteon from scratch, because if you can draw it, you can answer anything about it. Picture a tree stump kind of here with its growth rings. These are the rings, 1, 2, and it keeps going like this. And now drill a pipe straight down the center here. That central pipe is the Haversian or central canal, and it carries blood vessels and nerve along the bone axis. That is the lifeline. Every cell in this osteon depends on it. Around that pipe, the three rings are the concentric lamellae. One concentric lamellae, another one, and it keeps moving in like this. In other words, we can say that concentric lamellae are rings of mineralized matrix around the central canal. Concentric just means sharing the same center, like the rings of an onion. Now, living cells are trapped between those rings. You see the blue star-like structures? Those are the cells. The little caves they live in are lacunae. So lacunae are the small cavities between lamellae, each one of them housing one osteocyte. So this osteocyte is inside the lacunae. One over here, one here. There are numerous osteocytes shown in the diagram. But a cell sealed in solid rock would starve, so nature drilled tiny tunnels. Those are the canaliculi. You see here, another one here. These canaliculi are connecting one osteocyte with another. In other words, we can say that canaliculi are microscopic channels radiating from lacunae that link osteocytes to each other and to the vessels. Picture a spider web of hair-thin tunnels connecting every cave to its neighbors and back to the central pipe. So food and signals can pass from the blood vessel, cell to cell, all the way out. That is how a cell buried in stone always stays alive. Now, if we zoom out a little and see on the left side of our presentation, you will see Volkmann's canal. This Volkmann's canal, you see here, they are cutting across vertical osteons. One osteon here, another here. And they are cutting across the osteons at right angles. That means they are perpendicular, and they connect adjacent osteons and reach the periosteum and endosteum. One osteon here, another one here. Both of them are being connected by Volkmann's canal here. I'm not referring to the blood vessels. Blood vessels are separate, but there's a canal that is called Volkmann's canal, and that stays perpendicular to the Haversian canal. If Haversian canals run up and down the bone, up and down like this, Volkmann canal runs sideways, linking one osteon pipe to the next and connecting the whole network to the blood supply on the surfaces. Between fully formed osteons, you will also see leftover fragments, the interstitial lamellae, which are irregular remnants of old osteons partially resorbed during remodeling. The demolished ruins of osteons that were torn down and rebuilt over the years. And finally, wrapping the whole bone just under its surfaces, inside and out. You see here, if I point with my laser, these are the circumferential lamellae, parallel rings just beneath the periosteum and endosteum, like the outermost bark layer running around the entire trunk. Short break. What you're looking at is the written version of this course, the complete IMAT Biology Guide, 19 full lectures. Every trap that cost students points marked, every pearl boxed, comparison tables where the exam likes to confuse you, and IMAT-style questions with the full reasoning worked out. Not just an answer letter. It is pay what you want, $15 minimum, purchasing pair parity enabled. And every future update is free for life. The link is in the description and in the pinned comment. Now back to the video. Moving to the anchor slide, we just will match what we have read. First of all, we have said compact bone is dense and it contributes to about 80% of skeletal mass, and the structural unit is osteon that has been covered. In case of spongy bone, the structural unit is trabecular. And mechanism, we have perfectly covered those. Even after that, let's mark a few of the important words again. In case of Haversian canal, they carry the blood vessels and nerves along the bone axis. Concentric lamellae, Those are rings of mineralized matrix around the central canal. And lacunae are small cavities which house osteocyte. Canaliculi are microscopic channels radiating from lacunae which link one osteocyte to another one. In case of Bachmann canal, they run perpendicular, connect adjacent osteons and reach periosteum and endosteum. Interstitial lamellae, those are irregular remnants of old osteons, partially resorbed during remodeling. Resorb means broken down. Circumferential lamellae, those are parallel rings just beneath the periosteum and endosteum. We will reinforce our knowledge by looking at the comparison table. Now read the two architectures across the table so not a single cell is left unspoken. Structural unit. In compact bone, it is the osteon, also called the Haversian system. In spongy bone, it is trabeculi with no osteons at all. To get a clear picture, we have brought in this diagram. You see in the periphery, the whole area is made up of compact bone. But when you keep coming toward the center, you see trabeculi, and those are the parts of spongy bone. And notice it does not have any osteon in it. Don't ever think that compact bone and spongy bones stay separately. In actuality, spongy bone and compact bone stay together as single unit. That means the bone has mixed structure, or our skeleton consists both of compact bone and spongy bone. When it comes to porosity, compact bone is low, 5 to 10% empty space. Spongy is high, 50 to 90% open. Nutrient supply. Compact is fed by Haversian canal blood vessels, threaded through those central pipes. Spongy is fed by diffusion from marrow vasculature, the blood in the marrow soaking nutrients into the nearby struts. Location. Compact forms the diaphysis wall and the outer shell of the bone. Spongy fills the epiphysis and the interior of the flat, short, and irregular bone. Osteocyte housing. In compact, cells sit in lacunae within the concentric lamellae. In spongy, they sit in lacunae within the trabeculae. And main mechanical role, if we consider, compact resists bending and torsion, the twisting and flexing of a shaft. Spongy absorbs compression and houses the marrow, cushioning the joint ends while holding the blood factory. First trap, and it is a big one. Osteons and Haversian canals exist only in compact bone. Point to be noted, they will be found only in compact bone. Spongy bone is organized into trabeculi. Students see the word bone and assume osteons are everywhere. They're not. Why does spongy bone not need osteons? Because osteocytes in trabeculi receive nutrients by diffusion from blood vessels in the surrounding marrow, not from a central canal. The struts are thin enough and bathed in marrow blood, so nutrients simply soak in. There is no need for the elaborate central pipe delivery system that thick compact bone requires. So if a question asks where Haversian systems are found, the answer is compact bone only. Spongy bone, no osteons ever. Second trap, and this one shows up on image question. Bachmann canals cross at right angles to Haversian canals and are not surrounded by concentric lamellae. You can look at this image. This part shows Haversian system and this canal is Bachmann's canal. You will notice Bachmann's canal does not have any surrounding concentric lamellae, whereas Haversian system does it. 1, 2, you can keep counting. And the absence of concentric rings is exactly how you tell them apart in the cross-section image that we have seen. A canal with onion rings around it, remember, is Haversian canal. A canal cutting across with no rings of its own is Bachmann's canal. And one more distinction packed in here. Interstitial lamellae are remnants of old resorbed osteons, not newly deposited bone. They are ruins, not fresh construction. So on a labeled micrograph, ringed and running with the shaft means Haversian, Unringed and cutting across means vacuum. Irregular scrapes wedged between whole osteons mean interstitial. And here's the pearl that turns spongy bone from a boring honeycomb into a piece of brilliant engineering. Trabeculi orient precisely along the lines of mechanical stress, maximizing strength for minimum mass. The little struts are not random. You see over here. They line up exactly along the directions that force travels through the bone, like the diagonal braces in a bridge or a crane or you see in Eiffel Tower. You can compare these structures with the trabeculi, and these trabeculi are needed so the bone is strong without being heavy, and that engineering predicts its own failure. Disuse, whether from long bed rest or from the weightlessness of spaceflight, means the loading signal is lost, and the result is rapid trabecular bone loss. If the struts exist because of stress, then remove the stress and the body stops maintaining them. Astronauts and bedridden patients lose bone quickly for exactly this reason. Bone is a tenant that builds only when the rent, that means mechanical stress, is being paid. Here's a question that unlocks every bone disease in this lecture. The question is, why is bone both hard but not brittle? A brick is hard but shatters. A rope is tough but floppy. Bone somehow manages to be both stiff and shatter resistant at once. The answer is that bone is a composite material, approximately 35% organic matrix and 65% inorganic mineral. Roughly 1/3 soft stuff, 2/3 hard stuff blended together. Two ingredients, two opposite properties combined into one material that beats either ingredient alone. Understand this blend and the disease at the end of the lecture will feel obvious. Some of my viewers might think that there are ropes inside bones. Actually, no. I'm just trying to make a schematic diagram to explain how it might relate to our real life examples. So, first of all, the organic component. The organic component is called osteoid, and it is mostly type 1 collagen, about 90% of the osteoid, plus osteocalcin. So we can write type 1 collagen plus osteocalcin. And you can mention two other stuffs. One is osteopontin and proteoglycans. Collagen is a rope-like protein. That's why we have shown ropes over here. And this collagen is the same family of fiber that makes tendons and ligaments tough. What does collagen give the bone? It provides tensile strength, resistance to being pulled apart. Here is the proof, and it is a classic demonstration. Bone with mineral removed is flexible like a tendon. If you soak a bone in acid to dissolve away all its mineral, what is left is the collagen framework, and it becomes so rubbery you can tie it in a knot. It bends, it flexes, but it will not tear. That is tensile strength. Now the opposite ingredient, the inorganic part, and this inorganic component is hydroxyapatite crystals with the formula Ca10PO4,6OH2. And this hydroxyapatite is deposited within and around the collagen fibrils. The stone-like structures represent the inorganic minerals. This hydroxyapatite is a calcium phosphate crystal, and it packs into and around the collagen ropes like cement poured around steel. What does the mineral give? It provides compressive strength and rigidity, resistance to being crushed. And again, the proof by opposite. Bone with collagen removed is brittle like chalk. If instead you burn a bone to destroy the collagen and leave only the mineral, what remains holds its shape but shatters at the lightest step. exactly like a stick of chalk. So mineral resist crushing but cannot survive a bend. Now put them together and feel why nature chose a composite. The combination principle is this. Collagen will resist tension. Hydroxyapatite resists compression, and together they are stronger than either alone. The soft ingredient stops the bone shattering, the hard ingredient stops it bending. Each one covers the other one's weakness. And the analogy that makes this unforgettable is reinforced concrete, where the steel rebar is the collagen and the concrete is the mineral. Concrete alone cracks under bending. Steel alone bends under load. But steel bars embedded in concrete give you a beam that neither shatters nor bends, which is why every bridge and building on earth is built this way. Your bones invented the trick first. So the two components, the organic and inorganic ones, are not a list to memorize. They are a cause and effect machine. Tell me which ingredient is missing and I can tell you exactly how the bone will fail. No mineral, it will bend like a rubber. And if no collagen, it shatters like a chalk. Keep that machine running as we meet the diseases, because each disease is just one ingredient going wrong. The trap here is a subtle one about a single word, and that is osteoid. Osteoid is the unmineralized organic matrix secreted by osteoblast. It becomes bony only after hydroxyapatite deposition. In other words, osteoid is the soft rope framework laid down first before any cement is poured. It is bone to be, not bone yet. And pouring the cement mineralization requires adequate calcium, phosphate, and vitamin D, plus an enzyme called alkaline phosphatase. And this alkaline phosphatase is released in the osteoblast matrix vesicle. That means alkaline phosphate is released by osteoblast. Now watch the consequence, because it names two diseases at once. Vitamin D deficiency leads to osteoid that accumulates but fails to mineralize, producing rickets in children and osteomalacia in adults. Without vitamin D, the body lays down the soft framework but cannot harden it. So the bones stay soft, and that softness is rickets in growing child and osteomalacia in a grown adult. Soft unmineralized matrix piling up is the signature of that whole problem. And now the pearl that keeps two diseases from ever blurring together in your mind, because the exam deliberately parks them next to each other. First, osteomalacia and rickets are a quality problem. There is a normal quantity of matrix, but the mineralization is defective. The framework is all there, it just never hardened. So the bones are soft, the enzyme ALP is elevated because frustrated osteoblasts are working overtime and serum calcium is low or low normal, since the missing vitamin D was what let you absorb calcium in the first place. Contrast that with osteoporosis. You can look at the image here, and to be more precise, you can look at this one as well. And this osteoporosis is a quantity problem. There is a reduced quantity of matrix that is written here, but what little remains is normally mineralized. The bone is properly hardened, there is just too little of it. So serum calcium and ALP are typically normal. Same fragile bones at the bedside, completely different machines underneath. One is soft bone of normal amount, the other is normal bone of reduced amount. Different mechanism, different laboratory picture, different treatment. We will prove all of this on the pathology slide, but plan the contrast now. Malacia is soft, porosis is scarce. I've come to the anchor slide again just to show you guys that I have discussed every single important topic regarding this. Some people might struggle a bit to follow along the diagram only. That's why I always show the anchor slides along with the diagrams. So as you can see, we have covered the component parts, and we have covered the trap as well. Osteoid is not a bone, and these are discussed in details already. That osteoid is unmineralized. It has been capitalized because this is the most important keyword. And in case of mineralization, if osteoid wants to be real bone, then it requires calcium, phosphate, and vitamin D, plus an enzyme called alkaline phosphatase, which has diagnostic value in differentiating the rickets or osteomalacia with osteoporosis. And when vitamin D deficiency occurs, we have already said, it causes rickets in children and osteomalacia in adults. And the pearl section, osteomalacia is not osteoporosis, and I have already said osteomalacia means defective mineralization, and osteoporosis mineralization is normal, but the matrix quantity has been reduced, and you can differentiate it in lab value with ALP. In case of osteomalacia, the ALP is elevated, but in case of osteoporosis, ALP are typically normal. And with that, we complete the bone matrix composition and related pathology with it. Now we move to bone cells, and this is the heart of the lecture. The slide shows where the most marks are won and lost. So we slow right down here. Bone is a construction site that never closes. And like any construction site, it has a crew. There are 4 bone cells types. And here is the single most important fact about their family tree. 3 arise from mesenchyme, 1 from the hematopoietic monocyte lineage. Let me translate. Mesenchyme is embryonic connective tissue, the raw material that becomes bone, cartilage, fat, and muscle. 3 of our 4 cells, the builders, come from that connective tissue family. But the 4th cell, the one that demolishes bone, comes from a completely different family, the hematopoietic monocyte lineage. The same blood cell family that gives rise to your immune system's macrophages. Hold on to that split, because it is the #1 trap on this entire topic. The builder and the demolisher are not cousins. They come from different worlds. Let us meet all 4 across the table, 1 row at a time. First, the osteoprogenitor cells, or osteogenic cell, sometimes called stem cell. Its origin is the mesenchymal stem cell, as we have seen in the image. Its function, it is a mitotic stem cell that differentiates into osteoblast, meaning it can still divide, and its destiny is to mature into a builder. You find it located in the periosteum and endosteum, those 2 living linings we met earlier. Think of it as the apprentice waiting to become a bricklayer. Second, the osteoblast, the builder itself. That's why written forms bone matrix. Origin again, the mesenchymal stem cell. Its function, it secretes osteoid and initiates mineralization. That is, it lays down the collagen framework, then starts hardening it. Exactly the osteoid then mineral story from the last slide. Its key markers are ALP, abundant rough endoplasmic reticulum and Golgi apparatus, and a single nucleus. That abundant rough endoplasmic reticulum and Golgi apparatus is a giant clue, because those are the organelles a cell uses to manufacture and export protein. And collagen is a protein. So a plump cell stuffed with protein export machinery is a builder. That you see here, it's kind of plump. Then we have said stuffed with protein export machinery. That means you will have rough endoplasmic reticulum scattered here, 1 single nucleus here. And that actually signals a builder. Third, the osteocyte. You can see in the image, this is the osteocyte. Written maintains bone tissue. Its origin is different and lovely. It is an entrapped osteoblast. When a builder lays down so much matrix that it walls itself in, it retires and becomes an osteocyte. A bricklayer who bricked himself into the wall. Its function? It's a mechanosensor that maintains the pericellular matrix, a sensor that feels mechanical stress and looks after the bone immediately around it. Its features are it lives in lacunae connected by canaliculi, those caves and tunnels from the osteon slide, and it is the most numerous bone cell of all. The retired walled-in caretaker is the commonest cell in bone. Fourth, the osteoclast, written resorbs bone. So we call osteoclast the demolition crew and the odd one out in the table. Its origin is the hematopoietic monocyte macrophage lineage, the blood and immune family, not the connective tissue family. Its function, it resorbs bone, dissolving both mineral and matrix, tearing the wall back down, cement and rope together. Its features are unmistakable. Trap as its marker. It is multinucleated. It has ruffled border. We can see again, it has ruffled border and it is multinucleated, as you can see. Why it is multinucleated? Because osteoclasts form by several monocytes fusing together into one giant demolition machine. No other bone cell has more than one nucleus, so multinucleation is a dead giveaway. Now a little more depth on the two cells the exam probes hardest. The osteoblast is a plump cuboidal to columnar cell with a secretory profile, abundant rough endoplasmic reticulum, and a large Golgi, and it releases matrix vesicles carrying alkaline phosphatase and calcium and phosphate. Picture a fat little factory. It pumps out collagen from its rough endoplasmic reticulum, and it also ships out tiny bubbles, matrix vesicles loaded with enzyme and the minerals needed to start hardening the fresh osteoid. The osteocyte, by contrast, has reduced organelles because a retired cell no longer manufactures much, but it grows long cytoplasmic processes through the canaliculi that form a body-wide mechanosensory network. Those long arms reaching through the tunnels let osteocytes feel strain across the whole bone and talk to one another. And critically, osteocyte apoptosis triggers targeted remodeling. When an osteocyte dies, its death is distress flare that tells the crew to come remodel the exact spot. Finally, note perilacunar remodeling. Osteocyte can dissolve small amounts of pericellular mineral under parathyroid hormone stimulation, providing rapid calcium release. So even the quiet caretaker can release a little calcium fast when the hormone PTH demands it. Another withdrawal from our calcium bank. These are the key features that we have covered just now, and I'm not repeating it because I have described every one of them under osteoblast image and osteocyte image. Here is the single most common error in this entire topic, and I'm going to hand you the fix. Osteoblast builds bone. Osteoclast resorbs it or breaks it down. The names sound almost identical, and that is exactly why students swap them under pressure. Here is my memory hook. A blast is a burst of construction, and a clast, as in iconoclast, is a breaker. Blast builds, clast crushes. Reinforce it with the family tree we keep returning to. Different lineages. Osteoblast from mesenchyme, osteoclast from hematopoietic monocyte precursors. They are not related cell types. Reinforce it again with the markers. Different markers such as ALP marks formation. So that is the osteoblast. And trap marks resorption. So that is osteoclast. And reinforce it one last time with a structural fact you can spot on any image. Osteoclasts are the only multinucleated bone cell containing anywhere from 2 to 50 nuclei. So if you see a giant cell with many nuclei chewing a bit on bone, you never have to guess. That is the osteoclast, the breaker. Get this pair right and you have secured the most heavily tested distinction in the topic. And the exam pearl gives you a skill. not just a fact, because the IMAT often refuses to name the cell and instead describes it, expecting you to identify it. So learn to read the cell from its organelles. A described cell with abundant rough endoplasmic reticulum and a prominent Golgi is a secretory cell, a protein exporter, so it is an osteoblast, a builder churning out collagen. A described giant multinucleated cell with a deeply folded membrane sitting in a surface pit is an osteoclast in a hoe-shaped lacuna, the breaker parked in the little crater it has dug. Remember, IMAT frequently describes the cell rather than naming it. So when a stem says a large cell with many nuclei and a ruffled membrane, do not wait for the word osteoclast. You have already been told. Read the description, picture the organelles, name the cell. That skill turns wordy trap questions into gifts. We just met the osteoclast, the demolition crew. Now let us watch it actually work, because how it dissolves rock hard bone is both clever and heavily tested. Remember what bone is made of. 2/3 of bone, hard mineral, and 1/3 of bone is soft collagen. To demolish bone, you must defeat both. And Osteoclast does it in 2 stages in order. And stage 1 or step 1 is sealing and demineralization. First of all, the osteoclast seals the bone via an actin sealing zone, creating an isolated compartment. In this image, you can picture a plunger. This cell looks like a plunger here, which is clamping down onto the bone surface. This is the bone surface, making a sealed pocket underneath, walled off from the outside, so whatever it releases stays concentrated. And inside that seal, the membrane facing the bone is thrown into deep folds. And for that, we're going to look the ruffled border, a deeply folded membrane that hugely increases the secretory surface area. As you see in this image, this ruffled border gives it a vast area to pump chemicals through, like the microvilli lining your gut. And after that, the pumping begins. Proton pumps drive hydrogen ion into the sealed space. That means this place. And as a result, the pH in there will drop to about 4.5. Hydrogen ions make acid. So the osteoclast is squirting acid into a sealed pocket against the bone, bringing it to a pH of about 4.5, roughly as acidic as vinegar. And what does acid do to the calcium phosphate mineral? Acid dissolves the hydroxyapatite, and calcium and phosphate are released. The cement dissolves away into the pocket, freeing its calcium and phosphate. And by that, stage 1 or step 1 is completed and the mineral is gone. But dissolving the cement leaves the collagen ropes still there. So stage 2 is matrix degradation. Now, lysosomal enzymes are secreted into the acidic compartment. That means they were being secreted over here. Lysosomal enzymes are protein digesting scissors. And the star enzyme is cathepsin K, a cysteine protease which cleaves type 1 collagen and other osteoid proteins. Cathepsin K cuts the collagen ropes to pieces in that same acidic pocket. And to demonstrate that, we show this picture where you see the ropes are getting broken down. With mineral dissolved and collagen chopped, the bone at that spot is fully demolished, and the little crater left behind on the bone surface has a name. You see the crater over here? All the contents are broken down and dissolved, and the crater that is left is called hoe-shaped lacuna, that is written here. So the whole demolition is, first would be clamp and seal, then acidify to dissolve mineral, then enzymes to chew collagen, which will eventually leave us with hoe-shaped lacuna. So acid first, enzymes second, and the order will be always in this fashion. What we have seen in the image, we need to match that with the slide. So we have covered that osteoclasts seals to bone via an actin sealing zone, creating an isolated compartment. Then ruffled border usually increases secretory surface area. The structure that looked like the villi of intestine. Then proton pump drives proton into the seal space, and as a result pH drops to about 4.5, and acid in there will dissolve the hydroxyapatite, and as a result calcium and phosphate will be released. Step 1 done, and in step 2 the remaining matrix will be degraded. That's why it's called matrix degradation. Lysosomal enzymes which are secreted into the compartment will cleave type 1 collagen and other osteoid proteins. And one famous example is cathepsin K, which breaks down the type 1 collagen and other osteoid protein. And the remaining pit or the excavated pit on the bone surface is called Haversian lacuna. A demolition crew this powerful cannot be allowed to run wild, so there is a control system. And it contains one of the most beautiful and most tested ideas in bone biology. And here's the twist that surprises everyone. Osteoblasts, the builder, control the breaker. The foreman controls the wrecking crew. Let me build the 3 players in our canvas face, then we will come back to our anchor to match what we have read. First, the osteoblasts will express RANKL or RANKL on their surface. You see, these are the RANKL, and RANKL is a signal molecule, a green light displayed by the builder. Second, the RANKL will bind to the rank on the osteoclast precursor. That means RANKL is part of osteoblast, and you see over here, this is rank, which is part of osteoclast, or osteoclast precursor to be more precise. When the RANKL binds rank on the osteoclast precursor, this drives differentiation into an active osteoclast. That means the osteoclast precursor will become an active osteoclast after this binding. So it's pretty obvious that rank is the receiver on the young osteoclast. When the builder's green light, rank L, plugs into the breaker's receiver rank, the breaker matures and switches on. So the builder literally activates the breaker by showing it a green light. But now the system needs an off switch, and here it is. Elegant and simple. Third, osteoblasts also secrete OPG or osteoprogenitor, a soluble decoy receptor. OPG is a floating fake receiver. What does a decoy do? OPG will bind rank L. That means they will bind to the rank L, which will block the rank L from reaching rank, and osteoclast activation is inhibited. The decoy grabs the green light molecule out of the air before they can reach the breaker. That means these OPG molecules don't allow the connection of rank and rank L, and consequently the breaker or the osteoclast never gets switched on. That means the same builder or osteoblast makes both the accelerator, which is rank L, and the brake, which is OPG. And the master conclusion, the sentence to memorize, is that the rank L to OPG ratio sets the rate of bone resorption. More rank L relative to OPG. That means more green lights get through, and that will mean more demolition. On the other hand, when more OPG relative to rank L, more green lights get intercepted. That means less demolition. That single ratio controls how fast your bone is torn down, and it is the lever that hormones and drugs and menopause all pull. Now we switch to the anchor phase again to match our description of the canvas phase. Osteoblast will express rank L. We completed that. Then rank L binds rank on the osteoclast, done as well. And when this binding happens, the osteoclast precursor will become osteoclast or active osteoclast. When it is time to stop, osteoblast will secrete OPG or osteoprogenitor, which is a decoy receptor, and OPG will bind to rank L, which blocks it reaching rank. As a result, osteoclast activation will be inhibited. So rank and OPG ratio, the final part, is the metric or the factor that will set the rate of bone resorption. Now the trap this control system sets, and it is a favorite. You would assume that PTH, or the parathyroid hormone, which is responsible for breaking down bone, will act directly on osteoclast. Actually, it doesn't. PTH receptors, or PTH1R, actually sit on osteoblast. Read that twice. The hormone that drives down bone breakdown lands on the builder, not the breaker. Why? Because PTH makes osteoblasts upregulate RANKL, which then activates osteoclasts indirectly. That means the pathway is osteoblast mediated. PTH tells the foreman to show more green lights, and the foreman switches on the wrecking crew. The breaker never hears from PTH directly, it only obeys the builder. Now contrast this with the opposite hormone, which is calcitonin. Calcitonin receptors are on osteoclasts, so calcitonin inhibits them directly. Calcitonin, the hormone that stops bone breakdown, does land straight on the breaker and it shuts the breaker off. So the pattern to hold is a beautiful asymmetry. PTH works on bone breakdown indirectly through the osteoblast. Calcitonin works on it directly on the osteoclast. Mixing those up is one of the most reliably punished mistakes in the whole topic. And here's a genuinely high difficulty pearl that separates top scorers. The teriparatide paradox. Continuous PTH exposure leads to increased resorption and net bone loss. Exactly what you see in disease hyperparathyroidism, where the gland pumps out PTH non-stop and the skeleton is slowly eaten. That fits everything we just said. But now the twist. Intermittent low-dose PTH given as the drug teriparatide leads to net bone formation. The very same hormone given in short daily pulses instead of a constant flood builds bone up. Same molecule, opposite outcome, decided entirely by the pattern of exposure. Steady versus pulsed. That's why written same hormone, opposite skeletal outcome decided by the pattern of exposure. You do not need the deep molecular reason for the IMAT. But you must know the headline. Constant PTH destroys bone, pulsed PTH builds it. When a hard question tries to catch you by saying PTH is used as a drug to build bone, you will know it is not a misprint. It is the teriparatide paradox. Now we ask where bone actually comes from in the first place, how it is born in the embryo, and there are two completely different methods. And this slide covers the simpler, more direct one. Intramembranous ossification is the direct formation of bone here from mesenchymal connective tissue with no cartilage intermediate. Remember mesenchyme? Mesenchyme is the raw embryonic connective tissue. In this method, the raw tissue turns straight into bone with no rehearsal, no template. Contrast that with the method on the next slide, where the body first builds a full cartilage model and then replaces it. Here there is no model at all. The bone forms directly inside a sheet of connective tissue, which is why it is called intramembranous, meaning within a membrane. Now we will walk the steps in order. The first step would be the mesenchymal cells you see here condense in a highly vascularized region of the embryonic connective tissue. You see the mesenchymal cells are gathering particularly around this place. These raw cells will cluster there because there's a spot which is rich with blood vessels, and the crowding is the trigger. After that, the step 2 is also seen in this image. In step 2, the condensed cell, that means the mesenchymal cells, differentiate directly into osteoblast. These are osteoblasts that you see, forming an ossification center within the membrane. The crowded raw cells become builders on the spot, and that starting point is called an ossification center, the seed crystal from which bone will spread. Step 3, those osteoblasts will secrete osteoid, the little dots you see, and these osteoids will rapidly mineralize into woven bone. The builders will lay down collagen framework and harden it fast into a rough first draft bone called woven bone. In step 4, blood vessels will invade. You see over here. These are the blood vessels. And osteoprogenitor cells will arrive and the woven bone is remodeled into mature lamellar bone. Reinforcements and vessels move in and the rough draft gets rebuilt into neat layered mature bone. And step 5, peripheral mesenchyme condenses to form the periosteum. So the leftover raw tissue around the edge becomes the living wrapper we met earlier. This is the wrap. So we can say it again in this fashion is that the mesenchymal cells will crowd, then it will convert to the builders or osteoblasts, then there will be lay down of rough bone, then it will upgrade to mature bone. Finally, we will wrap it and that's done. This whole diagram is included in my guide. If you want, then you can look at my guide to get the full picture. So we can review the sequence again in our anchor slide. First is mesenchymal cells will condense in vascularized region. Then condensed cell will become osteoblast. Osteoblasts will secrete osteoid, which rapidly mineralizes into woven bone. Blood vessels will invade, osteoprogenitor cells will arrive, and the woven bone will become lamellar bone. Finally, peripheral mesenchyme condenses to form periosteum. So which bones are built this quick, direct way? The flat bones of the skull vault, the frontal, parietal, temporal, and occipital bones. That means the flat plates of your cranium. So just a quick look at the flat bones. We said frontal. This is the frontal bone, then parietal bone, temporal bone, and occipital bones in the back. Then mandible, we have seen the picture already. This is the bone of our lower jaw. It is also made or ossified in intramembranous ossification process. The clavicle, the collarbone. And remember, we flagged clavicle 2 slides ago for a surprise. Here it is. That means clavicle might look tricky, but you must remember it is formed by intramembranous ossification. And parts of the skeleton, or to be precise, parts of the facial skeleton also falls under intramembranous ossification. And you notice the theme here. The plate-like bones that need to form quickly to shield the brain, plus a couple of special cases. That flat plate theme is your memory anchor for this whole list. And here's the surprise, the trap we set up earlier. Students assume the clavicle, being a long thin bone, must form the long bone way, that means endochondraly, but it doesn't. The clavicle forms primarily by intramembranous ossification with only a minor endochondral contribution and its very end. That means these are the ends. It looks like a long bone, but it is built like a flat bone. 2 bonus facts the exam sometimes adds. It is also the first bone in the body to begin ossifying very early in the embryo and the last to complete ossification, not finishing until your mid-20s. So the clavicle is the exception that proves you cannot judge a bone's formation method by its shape. When a question lists bones and asks which forms intramembranously, the clavicle is the sneaky correct answer hiding among the obvious flat skull bones. So the trap is covered. That clavicle, being a long bone, should have been endochondral, but it is in actuality forms by intramembranous ossification with minor endochondral contribution at its end. It is the first bone to begin ossifying and the last to complete the ossification process. A second final trap about the skull itself. Do not say the skull forms intramembranously. Full stop. Because the skull is not one thing. The skull vault, seen on the left, the flat dome-shaped calvarial bones on top, forms by intramembranous ossification the direct way. But the skull base, the bones at the bottom that the brain sits on, seen on the left, right, forms by endochondral ossification, the cartilage model we cover next. Both belong to the same skull. The mechanisms differs by region, not by bone name. So remember, skull vault forms by intramembranous ossification, skull base forms by endochondral ossification. Both belong to the same skull, but mechanisms differ. So if a question asks about the base of the skull specifically, the answer flips to endochondral. Vault on top is direct, Base underneath is the long way around. And now let me share a memorable clinical pearl that follows straight from intramembranous skull formation. This pearl talks about fontanelles. Fontanelles are the wide, unossified gaps you see here. This part is unossified, this one is unossified too. These unossified fibrous gaps are called fontanelles, which are seen between the developing skull bones in an infant. You can feel these fontanelles as soft spots on baby's head. Because the flat skull plates form as separate islands within a membrane and only slowly grow toward each other, there are gaps of soft membrane between them at birth. Those gaps are not flaw, they are essential, and they do two jobs. First, they permit skull deformation during birth, letting the baby's head compress slightly to pass through the birth canal, and they accommodate rapid brain growth in the first year or two, when the brain is expanding first. For the exact numbers the exam may want, the anterior fontanelle, the big one on top that we see in the image, closes much earlier at roughly 2 to 3 months. Actually, the anterior one will close at 18 to 24 months. The posterior one will close at 2 to 3 months. So a slight correction from my side. So this slide is completely covered. We have covered all the traps and the pearl as well. Now we will discuss endochondral ossification, and after that epiphyseal plate. First, focus on endochondral ossification, which is the second one of the ossification process. And this is the more common way a bone is born. And this one is responsible for almost your entire skeleton. Endochondral ossification is the replacement of a pre-existing hyaline cartilage model with bone. Break the word down. Endo means within. Chondral means cartilage. So bone forming within cartilage. Here the body does not rush. It first sculpts a complete full-shape model for the future bone out of soft hyaline cartilage, like a sculpture clay maquette, and then slowly replaces that clay with real bone. This method forms most bones of the body, all the long bones, the short bones, the irregular bones, and the skull base. So the arms, the legs, the wrist and ankle bones, the vertebrae, and the base of the skull all come from this cartilage model route. If intramembranous was the quick sketch, endochondral is the careful sculpture. Here comes the mechanism or sequence of endochondral ossification. It has 7 steps and they tell a story. So follow the plot. Step 1 would be mesenchymal cells become chondroblasts and secrete a hyaline cartilage shaped like future bone. You see here. The raw cells become cartilage makers and build a clay model in the shape of the bone to be. Step 2, the central chondrocytes hypertrophy. Here, calcify the matrix and die. The calcified cartilage is a scaffold, not bone. So step 1 and step 2 complete. In the middle of the model, here, the cartilage cells swell up, harden the material around themselves with calcium and then die off, leaving a hardened but temporary scaffold. Say it clearly. Calcified cartilage is not bone, it's just a stiffened template. Step 3, the key invasion. A periosteal bud made of a nutrient artery, here, and osteoprogenitor cells plus osteoclasts invades this periosteal bud, which is a supply convoy, and it drills into the model. You see here, it is drilling here, which brings a blood vessel, builder stem cells, and demolition cells. Then the osteoclasts resorb the calcified cartilage while the osteoblasts deposit osteoid on the remaining spicules, creating the primary ossification center in the diaphysis or the shaft you see here. So what I've said, let me repeat it. Osteoclast will resorb the calcified cartilage and osteoblast will deposit osteoid, and it will create the ossification center, the primary one. The breakers or the osteoclasts clear away the temporary cartilage scaffold, and right behind them the builders lay real bone onto the leftover struts. The first true bone site in the shaft is the primary ossification center. So step 3 is complete. Now moving to step 4. Now osteoclast will excavate the medullary cavity, which fills with bone marrow. You see, we have osteoclasts excavating the center or the mid portion of the bone where bone marrow will fill out. So we can say, in other words, the breakers hollow out the center of the shaft to make the marrow space, which fills with the red blood making marrow. Now moving to step 5. After we are done with step 4, step 5, what's happening here? In this step, secondary ossification centers appear in the epiphysis, spreading outward from the center. Later, you see 2 secondary ossification centers shown here. So later what happens? Neutral bone sites ignite the 2 ends of the bone. Step 6, and this is the one that decides your height. The epiphyseal plate of the hyaline cartilage, you see here, these are the plates, persist between the primary and secondary centers, permitting longitudinal growth. Between the bone forming in the shaft and the bone forming in each end, a stripe of original cartilage is deliberately left behind. That is the epiphyseal plate. I'm repeating the information so that it becomes easy for you, because sometimes if I go very fast, then it's gonna be difficult for you to comprehend and you might lose focus. And as long as this epiphyseal plate or the cartilage stripe keeps growing, the bone keeps getting longer longitudinally. That leftover stripe, the growth plate, is very important. Although the annotation or the labeling is wrong, you need to remember the growth plates are here. These are the growth plates. Step 7, at skeletal maturity, the plate ossifies into epiphyseal line, you see here, and growth stops. When you finish puberty, that last cartilage stripe finally turns to bone, leaving the scar line we met earlier, and you cannot grow any taller. The whole story from start would be build a cartilage model, harden and clear its center, replace it with bone from the middle out, keep a cartilage stripe for growth, and finally close that stripe when growth is done. And with our canvas phase, we covered the whole of mechanism. Even after that, let me recap a bit so that it becomes permanent in your memory. First, mesenchymal cells become chondroblast, they secrete cartilage model, then central chondrocytes hypertrophy, that means their cell size becomes larger, they calcify the matrix, then finally they die. Then a periosteal bud, which contains the artery, osteoprogenitor cells and osteoclasts, invades the center of the bone. Then osteoclasts excavate the center of the bone to create medullary cavity, which eventually gets filled with red marrow. Then secondary ossification centers appear in the ends of the bones, that means epiphysis, spreading outward from the center. Epiphyseal plate of hyaline cartilage persists between primary and secondary centers, which permits longitudinal growth. And when puberty comes, the maturity completes and the epiphyseal growth plate disappears, you can say, and you can see a line as a remnant over there. Now zoom into that growth stripe itself, because the exam wants its layer in order. Read the table from the epiphysis site toward the diaphysis. Zone 1 would be reserve or resting cartilage. A small quiescent chondrocytes anchor the plate to the epiphysis, a calm anchoring layer. Zone 2, proliferating cartilage. Chondrocytes divide rapidly and stack into columns, you see here. And this is where growth actually occurs. The engine of lengthening, cell multiplying and piling into neat columns. Zone 3, hypertrophic cartilage. The chondrocytes enlarge and the matrix begins calcifying. The cells swell and the material starts to harden. Zone 4, calcified cartilage. The calcified matrix serves as the scaffold for bone deposition. And zone 5, Ossification. Osteoblasts deposit bone on the spicules while osteoclasts clear old calcified tissue, where cartilage finally becomes bone. And your memory sequence would be written here underneath: RPHCO. Reserve, P for proliferating, H for hypertrophic cartilage, C for calcified cartilage, O for ossification. So let me repeat again: RPHCO. From the end toward the shaft, and you own the growth plate. Now let's look at the first exam trap. The trap here catches two errors at once. First, the growth plate is hyaline cartilage, not fibrocartilage. When the exam asks what type of cartilage the epiphyseal plate is made of, the answer is hyaline, the same glassy cartilage that caps the joints, and structurally it counts as a joint in its own right, as synchondrosis, a type of cartilaginous joint we will formally meet later. Second, and this is the subtle one, articular cartilage at the bone ends is never replaced by bone in normal development. Only the plate ossifies. So even though endochondral ossification is all about replacing cartilage with bone, the thin cap of articular cartilage on the very joint surface is spared and stays cartilage for life. Only the internal growth plate closes, the joint cap remains. Mix those up and you will wrongly think that the joint surface turns to bone, which it never does. Now if we look at the pearl, we're going to notice the pearl ties the growth plate to hormones and to the clinic. Growth hormone does not act on the whole plate randomly. Growth hormone stimulates the liver to make IGF-1, insulin-like growth factor 1, which acts on the chondrocytes in the zone of proliferating cartilage, seen here. This is the zone of proliferating cartilage, which is the layer exactly we identified as the engine of growth. So the height-driving hormone targets the height-driving zone. That is satisfying and testable. Now the familiar consequence seen through this lens. Excess growth hormone before plate closure causes gigantism because the plate is still open and the whole body lengthens. Excess growth hormone after closure causes acromegaly. Let me write down here. Because the plate is shut, length is impossible and the only width remains. Same rule as before, now anchored in zone 2. And the clinical bonus the exam likes: a Salter-Harris fracture, a break that runs through the growth plate in a child, risks permanent growth arrest and limb length discrepancy, seen here in the infographic or the diagram. Because if you damage the proliferating zone, the bone may simply stop lengthening while the other side of the body keeps going. That is why fractures through a child's growth plate are treated with special seriousness. And with that, we covered the exam pro. Many students of me keep asking me about where to get these pearls and traps. I have actually released a free anchor slides doc where you can get all the lectures for free. You can check that out in the bio. And with that, we arrive at the slide 11, which talks about bone remodeling and fracture repair. Bone looks permanent, but it is one of the most restless tissues in your body. Remodeling is the lifelong replacement of old bone with new bone, roughly 10% of the adult skeleton per year. Think about that. About a tenth of your entire skeleton is torn down and rebuilt every single year. So that over roughly a decade, you have replaced almost your whole skeleton without noticing. It happens at discrete sites called basic multicellular units, or BMUs, which are little teams of breakers and builders working together at one spot. And the purpose is twofold. First is to repair fatigue microdamage and to respond to mechanical and mineral demands. It fixes the tiny cracks that everyday loading creates before they grow into a real fracture. And it lets the skeleton adapt, thickening where it is stressed and releasing calcium when the blood needs it. Bone is not a finished statue. It is a building under permanent renovation. Now we will understand the mechanism of the renovation in details in our canvas phase. The renovation follows 5 steps. You see here 1, 2, 3, 4, 5, and always in this order. Step 1 is activation. You would see that the lining cells here will retract, exposing the bone surface, and osteoclast precursors are recruited and they fuse here. The protective lining peels back and demolition crew assemble. Step 2. Resorption. Osteoclasts excavate a house-shaped lacuna over 2 to 4 weeks, seen over here. The breakers dig their pit, the same acid then enzyme demolition from slide 8 over a few weeks. Step 3 is reversal. The osteoclasts apoptose or migrate away, and mononuclear cells prepare the cavity floor. The breakers finish and leave, and the site is cleaned and primed for construction. Then comes step 4, formation. Osteoblasts fill the cavity with osteoid over several months. You see over here. Osteoid mineralizes into lamellar bone. The builders move in and slowly refill the pit with fresh, properly layered bone. And notice formation takes months while resorption only takes weeks, which is an important asymmetry. And then comes step 5, quiescence. Lining cells cover the surface until the next activation signal, and the site goes quiet until its next renovation. Now the crucial balance point. In a healthy individual, the amount broken down equals amount rebuilt. But when resorption exceeds formation, the result is net loss, or we can call it osteoporosis. And as the breakers outpace the builders, cycle after cycle, you slowly lose bone, and that running deficit is called osteoporosis. Keep that equation in mind. It is the whole disease in one line. Now we will talk about the fracture repair sequence, the relative process of healing a break, which reuses cells and even echoes embryonic bone formation. 4 stages you can see here. Stage 1, hematoma. A blood clot forms at the fracture, you see here, with an inflammatory response and cell death at the bone ends. First, there is bleeding and a clot, exactly like any wound, plus some inevitable cell death right at the broken surfaces. Stage 2 would be fibrocartilaginous or soft callus. Fibroblasts and chondroblasts build a fibrocartilage bridge across the gap. A soft, rubbery patch of fibrous tissue and cartilage spans the break. You see in this image, holding the ends loosely together, which you can consider as a temporary splint. Stage 3 would be bony or hard callus. Periosteal osteoblasts convert the soft callus to woven bone, you see in this segment. So what it means is that builders from the periosteum that prepares cell reservoir we keep praising replace the soft patch with rough woven bone, a firmer but still crude bridge. And stage 4 would be remodeling. Here, the woven bone is converted to lamellar bone, and the Haversian systems and the bone contour are restored. So that means that finally the rough repair is rebuilt into neat, mature bone with proper osteons, and the bone is filed back into its original shape. So the sequence would be, if I write again, clot, then soft bridge, then hard, rough bridge, and polished final bone. Now let's get familiar with one important exam trap. The trap concerns 2 words that keep appearing: woven and then lamellar. Woven bone, which has immature randomly arranged collagen, is always the first bone laid down in both fetal development and fracture repair. It is quick and rough, collagen thrown down in a disorganized weave. On the other hand, lamellar bone, with its mature, organized, and concentric layers, replaces it later during remodeling, which you see on the right side. It is the neat, layered, load-bearing final product. So the sequence is always woven first, lamellar second, whether you are an embryo building a skeleton or an adult healing a break. And the exam-relevant conclusion is fracture callus containing woven bone is a normal intermediate stage, not a pathological finding. If a question shows woven bone in a healing fracture and asks if that is abnormal, the answer is no. That is exactly what a healing bone should look like partway through. Woven is not diseased. It is just young bone that has not been upgraded yet. And a pearl about blood tests, which sets up the disease slide. Remember, ALP marks formation, or it talks about the osteoblasts, and TRAP marks resorption, or it marks the osteoclast. So we can write resorption. In fracture healing, in Paget disease, and in hyperparathyroidism, both ALP and TRAP are elevated together, that you see here, because in all three the whole renovation process is running fast. When both crews are working overtime, both their markers go up. So high everything means high turnover. Breakers and builders, both of them are busy here. And contrast that with osteoporosis, where the imbalance favors resorption, so the resorption markers rise relative to the formation marker. Here, the breakers are winning, so their marker leads. You see the tall column of TRAP? It signifies the osteoclast. So this lets you read the machinery from a blood panel. Both markers up together point to a high turnover state like healing or Paget disease, and when resorption leads, it points to net loss like osteoporosis. Hold that, because on the pathology slide, these blood values become the fingerprint that names each disease. Now we come to the hormonal regulation of bone and calcium. This is the control room where the whole lecture comes together, because every hormone here is really adjusting one thing, and that is the calcium in your blood using bone as the bank. 6 players. For each, learn 3 things. First, learn the stimulus that releases it, its effect on bone, and the direction it pushes blood calcium. Let me read the table row by row and make each one make sense. First comes the parathyroid hormone from parathyroid chief cells. Its stimulus is low serum calcium, so it fires when blood calcium drops. And what is the effect it will show? You see in this image, parathyroid hormone raises osteoblast rank L, you see here. To be more precise, you need to put here. You see so many rank L's here, and it activates the osteoclast indirectly here, which is the exact indirect pathway from the slide 8. The foreman, that means the osteoblast, tells the wrecking crew to work, and its direction, it raises blood calcium, making the withdrawal from the bank. Low calcium calls PTH. PTH breaks down bone, calcium rises. Secondly, we will talk about calcitonin from the thyroid parafollicular C cells, and its stimulus is the opposite, that is high serum calcium, and its effect, it directly inhibits osteoclast, you see here, which carry its receptor, shutting the breakers off. And remember, this is the direct acting hormone. Its direction, it will lower blood calcium. High calcium calls calcitonin, breakers stop, calcium fall. PTH and calcitonin are mirror images. That means calcitonin does the opposite of what PTH does. Let's match that information again with the table. Calcitonin comes from the parafollicular C cells of thyroid. Stimulus is high serum calcium, and it will inhibit osteoclast. As a result, the serum calcium is lowered. Serum calcium, you can just consider that as blood calcium. That means PTH will raise the calcium in serum, or you can tell it can cause hypercalcemia, and calcitonin will cause hypocalcemia. Third comes calcitriol, the active vitamin D from the kidney via the enzyme 1-alpha hydroxylase, and its stimulus is low blood calcium and parathyroid hormone. And what is the effect? It does 2 things. It permits mineralization of osteoid, letting bone harden properly, and its direction is to raise blood calcium, mainly via gut by boosting calcium absorption from food. To match with slide, you see calcitriol, the stimulus is low calcium and parathyroid hormone, and calcitriol or vitamin D is activated in kidney with the help of enzyme 1-alpha hydroxylase. Effect of bone permits mineralization of osteoid, and it raises serum calcium via gut. Now coming to the estrogen. Before we're going to look at the canvas phase, then we're going to talk about here estrogen, which is secreted from the testes and ovaries are released in tonic fashion, are released tonically, meaning steadily. Its effect is protective. It raises OPG, or osteoprogenitor, which suppresses osteoclast activation. That means it pumps out the brake molecule so the breakers stay quiet, giving a slight rise in calcium by preserving bone. That means it will raise calcium. Remember, estrogen protects bone. That single fact explains menopause bone loss. To match with the slide, you see estrogen, the source, testes and ovaries. It is released tonically, that means steadily, raises OPG, and it has slight rising effect on calcium. Fifth comes growth hormone and IGF-1. They are secreted from the pituitary gland and liver, and they are released on growth signals. What are their effects? They drive chondrocyte proliferation at the growth plate, the height engine from slide 10, with minimal direct effect on blood calcium. So that clears growth hormone part. Finally comes glucocorticoids. Glucocorticoids, also known as the stress steroids, which are secreted from the adrenal cortex, is secreted due to chronic stress or steroid therapy trigger. Their effect is harmful. They inhibit osteoblasts and reduce gut calcium absorption, starving the builders, and they lower calcium and thin the skeleton. That is why long-term steroid medication causes osteoporosis. Now we will discuss the mechanism of PTH. That says PTH has 3 actions in our canvas face, because without looking at the diagram, it will be a bit difficult for us to understand it. Remember, PTH is the star of calcium control. So let us see all 3 of its moves, because it raises blood calcium from 3 directions at once. Action 1. In bone, PTH hits the osteoblast PTH1R, and RANKL goes up, osteoclasts are activated, and calcium and phosphate are released from the dissolving bone. Action 2 is in the kidney. Tubular calcium reabsorption goes up. You see here that the tubules are absorbing calcium. As a result, less calcium is lost in urine, while phosphate reabsorption goes down, causing phosphaturia. That means phosphate is dumped into the urine. Hold that split. PTH keeps calcium but throws away phosphate. And action 3, also in the kidney, PTH stimulates 1-alpha hydroxylase, which makes calcitriol, which raises intestinal calcium absorption. You see here. So PTH also switches on active vitamin D, which pulls more calcium from your food. 3 fronts, 1 goal. And the goal is pull calcium out of the bone, save calcium in the kidney, and absorb more calcium from the gut. All 3 arrows point to higher blood calcium over here. So now we can understand the sequence here, that in bone it activates osteoclast, which causes calcium and phosphate to increase in blood. In kidney, the tubular cells reabsorbs the calcium, and as a result, calcium in urine is low, whereas phosphate in urine is high. Also in kidney, PTH stimulates 1-alpha hydroxylase, which activates vitamin D, and it causes, the vitamin D causes intestinal calcium absorption. And all 3 jobs to raise the calcium in blood. To understand the mechanism of vitamin D activation, we're going to go back to our canvas face, then we're going to review it from here. From our reading, we understood that the 3rd action of PTH involves calcitriol. So let us trace where active vitamin D actually comes from, because it is a 3-organ relay the exam loves. In the skin, UV light plus 7-dehydrocholesterol makes vitamin D3, cholecalciferol. Sunlight strikes a cholesterol-like molecule in your skin and makes raw vitamin D. In the liver, 25-hydroxylase converts it to 25-hydroxyvitamin D. That means the vitamin D3 that was made here, it comes to liver, then liver converts it to 25-hydroxyvitamin D. And the enzyme involved is 25-hydroxylase, and that is called calcidiol. This calcidiol is the storage form measured in blood. The liver actually does the first upgrade by converting vitamin D3 to 25-hydroxyvitamin D. And this half-activated form, the calcidiol, is what a blood test measures when checking your vitamin D level. Now, what happens in the kidney? Let's check. In kidney, as we mentioned earlier, 1-alpha-hydroxylase stimulated by PTH, written here, converts 1,2,5-dihydroxyvitamin D. That means, let me repeat it, the calcidiol comes to the kidney, then there is an enzyme in kidney called 1-alpha-hydroxylase. 1-alpha-hydroxylase will convert the calcidiol to 1,2,5-dihydroxyvitamin D, which is the active form of vitamin D. So we can say the kidney does the final activation, and notice PTH is what switches on that final step, linking the two mechanisms together. So the relay is skin makes it, liver stores it, kidney activates it under PTH command. Remember that the kidney is the activation organ, because when kidneys fail, this is exactly the step that breaks, as we will see on the disease slide that's written here. Now let's match the description with the slide, because some of my students struggle a bit to understand only looking at the canvas face. So we're going to match it again. So the sequence was in skin, sunlight and 7-dehydrocholesterol makes vitamin D3, which is called cholecalciferol. Then vitamin D3 comes to liver. There is an enzyme called 25-hydroxylase, and this enzyme converts vitamin D3 to 25-hydroxyvitamin D. And this is the storage form measured in blood. Finally, in kidney, there's an enzyme called 1-alpha-hydroxylase, which is stimulated by PTH. And this 1-alpha-hydroxylase converts the calcidiol to 1,2,5-dihydroxyvitamin D, which is also called calcitriol, and this is the final active form. Now we're going to look at the traps, and be careful because this traps is very confusing. And the trap is about the receptor pair, and it's worth saying slowly one more time because it is so heavily tested. The PTH receptor is on osteoblasts. PTH cannot act directly on osteoclast. The calcium raising hormone lands on the builder and works the breakers indirectly. The calcitonin receptor is on osteoclast in the second row that you see here written, and calcitonin acts directly and immediately on them. The calcium lowering hormone, that means calcitonin, lands straight on the breaker and shuts it off. So the asymmetry is PTH indirect through the builder, calcitonin direct on the breaker. Reversing these two is one of the most reliably punished error on this topic, which is precisely why I keep hammering it. If you remember nothing else about receptors, remember, PTH does not touch the osteoclast directly, it goes through the osteoblast. After the trap, now let's look at the pearl. And this pearl pays off the whole slide by explaining the most common bone disease on the earth in one clean chain. Estrogen falls at menopause. That means when the periods are over in case of female. And as a result, osteoblast OPG production will fail. Recall, estrogen's job was to raise OPG, the brake molecule. If you take the estrogen away, the brake production drops. Then less OPG means RANKL is unopposed. That means it is out of control now. And what is the result? Resorption exceeds formation, exactly the osteoporosis equation from the remodeling slide. And women can lose 3 to 5% of bone mass per year in the first 5 menopausal years. To be more precise, first 5 post-menopausal years, which is a startling rate. That means concerning. So, post-menopausal osteoporosis is not mysterious. It is a straight line from lost estrogen to lost OPG to unopposed RANKL to overactive breakers to net bone loss. Every link is something that we built earlier in this lecture, now assembled into a disease. That is exactly the multi-step reasoning the IMAT rewards. Now we leave hard bone for its softer cousin, which is cartilage. And this cartilage caps your joints, shapes your ears and nose, and cushions your spine. First, 3 properties that all cartilage shares and that explain most of its quirks. Cartilage is a specialized connective tissue that is avascular, alymphatic, and aneural. Break this down. Avascular means no blood vessels. Alymphatic means no lymph drainage. Aneural means no nerves. Because it has no blood supply of its own, it is nourished by diffusion from the perichondrium, or for articular cartilage, from synovial fluid. Nutrients have to soak in slowly from the covering membrane to the joint fluid, which is why cartilage heals so poorly. There is no bloodstream to rush in repair materials. And because it has no nerves, here is a striking consequence. The absence of nerves explains why articular cartilage damage can progress painlessly until late. Joint cartilage can be wearing away from years with no pain at all because there is nothing there to feel it until the damage reaches the bone underneath, which does have nerves. So no blood, no lymph, no nerve. Hold these 3. They explain everything. And there are 3 types of cartilage, each engineered for a different job. We need to read across the table. To match the points of this table, we're going to look at the diagrams so that it becomes permanent in our memory. So first one, collagen type. Hyaline cartilage. It has type 2 collagen fibers. Fibrocartilage has type 1 and type 2, and the extra type 1 being the tough tendon grade collagen. The elastic cartilage has type 2 plus elastin fibers. The elastin giving stretch. Now coming to perichondrium, the covering membrane. Hyaline has 1, but except on articular surfaces. Fibrocartilage has no perichondrium at all. Elastic always has 1. Appearance. Hyaline is glassy, translucent, blue-white, literally named for looking like glass. You see here. Fibrocartilage is opaque, fibrous white. Elastic is yellowish and highly flexible. Mechanical property. Hyaline is smooth with compressive strength. Fibrocartilage has tensile and compressive strength, the toughest of the 3. And elastic is flexible and returns to shape. Now key locations. Hyaline is at the articular surfaces, the epiphyseal plate, the costal cartilages, and the nasal, laryngeal, and tracheal rings. We know about them kind of in detail because we have covered them in respiratory system, and the costal cartilage is the cartilage that is attached to the ribs of chest, and fibrocartilage is in the intervertebral disc, the knee menisci, the pubic symphysis, the TMJ disc, and tendon insertion. TMJ means temporomandibular joint. And elastic is in the auricle of ear, the epiglottis, the external auditory canal, and eustachian tube. And regeneration is very limited for hyaline and limited for the other 2 because of that avascular nature. Match each type to its job. Hyaline for smooth, low friction surfaces. Fibrocartilage for heavy duty shock absorption. Elastic for things that must bend and spring back. So we have covered this whole table with specific diagrams so that we can print it in our memory. But you need to remember these keywords. You don't need to remember everything. But the key locations take important place in exams, so be careful about them. One structure deserves its own walkthrough because it is often tested, and the structure is called intervertebral disc. To get a clear picture, let's take a step back and go to our canvas phase. After that, we're going to complete this phase as well, or we're going to read the mechanism after it. Intervertebral disc is the cushion between 2 vertebrae, one over here, another one here, and between those 2 vertebrae, we see the intervertebral disc. And this intervertebral disc has 2 parts. First one is annulus fibrosus, the outer ring, and is fibrocartilage type 1 collagen arranged in concentric lamellae. You can imagine it like a tough layered ring like the wall of a tire. Then the nucleus pulposus. The inner core you see over here is a notochord remnant, a gel-like proteoglycan with type 2 collagen, a soft jelly center left over from an embryonic structure that we mentioned, and it is called notochord. So you can picture a jam donut, a tough ring on the outside here, soft jelly in the middle. Now the clinical event comes, and that is herniation. In herniation, the nucleus pulposus protrudes posterolaterally. Postero means behind, laterally means sideways. And it happens through a torn annulus, compressing the nerve root, and under strain, the jelly center can squeeze out through a tear in the ring. And because it bulges toward the back and side, that we said posterolaterally, it passes on nearby spinal nerve root, causing the classic slipped disc with pain shooting down a limb. So the summary is, if we talk about the intervertebral disc, then it's tough ring in the periphery, jelly in the center, and when jelly escapes, it escapes backward and pinches a nerve that you see over here. That is disc herniation. The first trap is about perichondrium. Most hyaline cartilage has a perichondrium, but articular cartilage is the exception because it has none. Why does that matter? Because it is nourished by synovial fluid instead, and it has no reservoir of progenitor cells, so it heals very poorly. Perichondrium is normally where repair cells live, and joint cartilage simply does not have that reserve. Round out the pattern. Fibrocartilage also lacks a perichondrium. Remember, two answers qualify: articular, hyaline, and all fibrocartilage, while elastic never lacks it. That perichondrium detail is a favorite discriminator. And the second trap is a knee mix-up. For that, we need to understand the meniscus or menisci. So the knee menisci are fibrocartilage pads sitting inside the joint. This one, C-shaped, is a meniscus. There is another meniscus on the right side. The menisci are there to improve congruity, the fit of the bones, and to absorb shock. And this is the catch, the articular surface of the femur and tibia, that means the blue shaded one, that is the articular surface, that is hyaline cartilage, not the meniscus. Students know the knee has fibrocartilage menisci and wrongly conclude the joint surface is fibrocartilage. It is not. So if a question asks what covers the articulating bone ends, the answer is hyaline, not fibrocartilage. The menisci are extra cushions wedged between the surfaces. The surfaces themselves are always hyaline. Two different cartilages in one joint doing two different jobs. So let me repeat it so that you never make a mistake. The knee menisci are fibrocartilage, but the articular surface are not fibrocartilage, they are hyaline cartilage. So keep that in mind always. And the clean single fact pearl, the epiglottis. This epiglottis is elastic cartilage, not hyaline, even though it sits right among the hyaline laryngeal cartilages such as thyroid, cricoid. Now you might ask why it is elastic. Because this structure, epiglottis, must bend repeatedly and spring back during swallowing, flapping down to cover your airway every time you swallow and popping back up after. And that endless bend and recoil requires elastin that I have written here. The auricle of the air is the other classic elastic cartilage example shown here for the same reason. Your outer ear bends and springs back. So among a list of throat cartilages, they are mostly hyaline. The epiglottis is the elastic odd one out, and the exam loves to plant it there. Epiglottis and air, the two body bendy springs, are elastic. That is written here. So let me repeat the remaining sentences. Epiglottis is elastic because it must bend repeatedly, and same for the auricle of the ear. Now in this section, we start bolting bones together into joints. A joint or articulation is any point where two or more bones meet. But not all joints are alike, and there are two ways to classify them, which the exam uses side by side. The first is structural classification based on what connects the bones: fibrous, cartilaginous and synovial. Is the glue fibrous tissue, cartilage, or a fluid-filled capsule? The second is functional classification based on how much they move. Synarthrosis means immovable. Amphiarthrosis means slightly movable. And diarthrosis means freely movable. So every joint has two labels, a structural one and a functional one. This slide covers the first two structural types, the ones that barely move. The freely moving synovial joints get their own slide next. Keep both classification systems running in parallel because a single question may give you one label and ask for the other. Now we will start with fibrous joints. This exact key features table has been pasted here with the diagram for easy comprehension. So fibrous joints are held by fibrous tissue and mostly do not move. There are three subtypes. Read across the table. First one is the suture, connected by a very thin fibrous layer between interlocking skull bones. You see the skull bone here. The sutures are the fibrous joint types. Functionally, the suture joints are synarthrosis, that means immovable, with examples being the coronal, sagittal, lambdoid sutures. These are different sutures of the skull bone. Second, the syndesmosis, connected by an interosseous membrane. You see here, this is the interosseous membrane that I'm pointing with my red ink. So whenever I say interosseous membrane, it means the sheets of tissue binding two leg bones or the two forearm bones, so they flex just a little as a pair. This is an example of forearm bone, to be precise. This is amphiarthrosis, as it can move slightly. And examples already has been mentioned, tibiofibular joint or radioulnar interosseous membrane. Third one, the gymphosis, connected by periodontal ligament. You see here in the image. And functionally, it is synarthrosis, with the example being a tooth in alveolar socket. That means, let's imagine this is a bone, it has socket in here, and when a tooth sits on it, the joint that is made is the gymphosis type of joint. And this is the only example of a gymphosis in the whole body. A tooth packed into our jaw is the one and only gymphosis. So if you see the word, think tooth immediately. So we covered the key features of fibrous joints seamlessly. Now we're going to move to cartilaginous joints. And for easy comprehension, once again, we're going to put it just beside or underneath our diagram. Cartilaginous joints are held by cartilage. There are two subtypes. First, the synchondrosis, the primary cartilaginous joint. The connector is hyaline cartilage. Functionally, a synarthrosis, that means immovable. And the examples are epiphyseal plate and the first sternocostal joint. One example given here, and this is the epiphyseal plate, which is an example of synchondrosis. And yes, interestingly, the growth plate, this growth plate itself counts as a joint, a synchondrosis, exactly as we flagged on the ossification slide. Second, the symphysis, the secondary cartilaginous joint. The connector is fibrocartilage with hyaline on each surface. Or to be more precise, we can say that the connector is fibrocartilage with a thin layer of hyaline on each surface. Point to be noted, a thin layer. So let's try to put that into perspective. This is a thin layer, another thin layer here, but in the center, there will be fibrocartilage. And functionally, it is considered amphiarthrosis. That means slightly movable, with examples of pubic symphysis, the intervertebral disc joints. So this example is intervertebral disc joint and pubic symphysis example given here. And the first one, that sternocostal joint, the example, if you zoom in, you're going to find it here. Sternocostal means the joint between the sternum and ribs. And notice the pattern. Synchondrosis uses hyaline and is immovable. That means synarthrosis. Symphysis uses fibrocartilage and moves a little. Hyaline for the rigid ones, tough fibrocartilage for the ones that flex under load, like the disc of your spine and the front of your pelvis. Now we will unravel the tracks one by one and finish this slide with the pearl. And let's bring this exam trap into our canvas face with some relevant diagrams. So the trap that catches many students is that the spine does not have just one kind of joint. It has two at every single level. The intervertebral disc joints seen here between vertebral bodies at the front. These are the vertebral bodies. And the bluish tinted joints are intervertebral disc joint. And these are symphysis written here. That intervertebral disc joints between vertebral bodies are symphysis. They are cartilaginous, made of fibrocartilage, and slightly movable. The cushions that we studied earlier. But the facet joints, also called zygapophyseal joints, between the bony arches, you see the bony arches in the back? These are the zygapophyseal joints. These joints are synovial. That is written that facet joints between vertebral arches are synovial. That means little plain joints with a genuine fluid-filled cavity. So the front of each vertebra connects by cartilaginous symphysis in the left one, and the back connects by synovial facet joints seen on the right diagram, and they are both at the same level. A question about the joints of vertebral column needs you to identify which one is meant. That means, does it mean the front one or it means the back one? If it says the joint between vertebral bodies, that is the symphysis. If it says the facet or zygapophyseal joint, that is synovial. Do not assume the spine is only one joint type. The second trap is one students walk straight past. They fail to classify the epiphyseal plate as a joint at all because it doesn't look like one, but it is a joint, and the joint is synchondrosis written here, also here. This diagram is actually visual depiction of our exam trap description, so you can look at both the trap text or the image. So as I was saying, the epiphyseal plate is synchondrosis, a hyaline cartilage joint, and functionally it is synarthrotic. That means immovable. Here is the twist that makes it interesting. It is a temporary joint that ossifies at skeletal maturity, becoming a synostosis, which just means a bony union, two bones fused into one. And the same fate awaits the skull sutures. You see the sutures? Sutures similarly convert to synostosis on old age. You see this? That means the jagged seams slowly fusing solid as you get older. So a synostosis is what a cartilaginous or fibrous joint becomes once it turns fully to bone. Growth plates and skull sutures are both temporary joints destined to become synostosis. If a question describes a former joint now fused to bone, that is synostosis. And now a memorable pearl that shows a symphysis actually earning its slightly movable label. During pregnancy, before we get into pregnancy, let's look at the left image and the right image. This is non-pregnant stage hip bone, but this one is in pregnancy stage. So during pregnancy, the hormone relaxin softens the fibrocartilage of the pubic symphysis. That means this, and sacroiliac joints. Those are here. These are the sacroiliac joints. That softening permits a slight separation of the pelvic ring, which will widen the pelvic canal. Actually, it will be birth canal, not pelvic canal. So it will widen the birth canal like this. And as a result of it, baby can pass through easily. Normally, the pubic symphysis, that means this, at the front of the pelvis barely moves. But under relaxin, it loosens just enough to give a few extra millimeters when they mirror most. It is the clearest example of a symphysis being functionally amphiarthrotic. That is written here, that it is the clearest example of symphysis being functionally amphiarthrotic rather than being fixed. So when the exam wants proof that a symphysis is not rigid, relaxin loosening the pubic symphysis in pregnancy is the answer. Now the joints you actually think of as joints, the freely moving ones that let you walk, grip, and turn your head. We're talking about synovial joints. A synovial joint is a freely movable joint, a diarthrosis with a fluid-filled joint cavity. You see in the image, this cavity has synovial fluid in it. And here's the defining feature. Let me repeat it. The one thing no other joint type has. The joint cavity lined by synovial membrane is the defining feature, unique to this joint class. Fibrous and cartilaginous joints glue the bones directly together with no gap. Synovial joints do the opposite. They leave a fluid-filled space between the bones. This fluid-filled space. And that space is what allows free movement. So the moment a question mentions a joint cavity or synovial fluid, you know you are dealing with a synovial joint. No other type has that gap. Let us build a synovial joint part by part. First comes the articular cartilage, and in the description it's written hyaline, smooth, avascular with no perichondrium, distributing load and reducing friction. The sleek cap on each bone end, that is the articular cartilage. Then comes the joint cavity. Joint cavity is the potential space you see here, which contains synovial fluid, and it is found in no other joint type. This is the defining gap. Then the articular capsule, an outer fibrous layer here, this whole surface, and this outer fibrous layer is continuous with the periosteum for stability, plus an inner synovial membrane for secretion. That means a two-layered sleeve wrapping the joint. Then comes the synovial fluid, shown here schematically. This is a plasma dialysate plus hyaluronic acid that lubricates, absorbs shock, and nourishes the articular cartilage, the slippery oil of the joint that also feeds the bloodless cartilage. Then comes ligaments. These are some examples of ligaments. These are bone-to-bone connections, which may be capsular or intracapsular, like ACL, PCO. These are anterior cruciate ligament, posterior cruciate ligament, or you can also say these are the ropes that stabilize the joint. Then menisci and articular disc. You see here menisci, and we have seen the disc previously in the images. These are fibrocartilage pads, and where menisci are C-shaped as in the knee and discs are complete in TMJ, the temporomandibular joint. That means the joint found in the mandible. Then sternoclavicular and acromioclavicular joints. So these are the extra cushions that improve the joint quality. Then bursae. You can see bursae over here. These are synovial sacs outside the joint that reduce friction where a tendon or skin crosses bone, like the subacromial and prepatellar bursae. These are little grease pads. And then tendon sheets. These are elongated bursae wrapping tendons in high friction zones of the hand, wrist, and foot. Sleeves that let tendons glide. That is the full anatomy of a freely moving joint. Don't worry too much about the detail. Just remember the names and you're good to go to pass the IMAT. Synovial joints come in 6 shapes, and it is classified by how many directions they move. Read the table. First one is plane or gliding. It's non-axial, gliding only, and examples are intercarpal, intertarsal, vertebral facet joints, etc. Those who can remember the examples, they will ace the exam, and if anyone finds the examples a bit difficult, they can skip it. But it's my duty to let you know about everything that can be important for IMAT. So as we have studied plane, let's look at the image. You see, this is a plane synovial joint, that is flat surfaces sliding over each other, depicted by this schema. Then comes hinge or gyngimus. It is uniaxial, movement is flexion and extension, and examples are the elbow at the humero-ulnar joint, the knee, the ankle, and interphalangeal joints. This hinge one is very important, and the movement is like the joint is moving in one plane like a door hinge. Then comes pivot or trochoid. It is uniaxial, it can rotate only. Examples are atlantoaxial, proximal radio-ulnar joints. Here you go, the pivot joint shown here. And this pivot joint, it spins around a single axis like shaking your head no. Then condyloid or ellipsoid joint. This is biaxial. Movement is flexion, extension plus abduction, adduction. And examples are radiocarpal, metacarpophalangeal joints. And they move in two planes. Here you see one condyloid joint. Then comes saddle. It is biaxial, like condyloid plus limited rotation is there. And with the classic example, the first carpo-metacarpal, that means thumb joint, and due to this reason your thumb is so dexterous. Saddle type is shown here. Then comes ball and socket. This is multiaxial. Movement is possible in all planes, and examples are glenohumeral, that means the shoulder joint, and coxofemoral, that means the hip joint. And this ball and socket type of joint is the freest joints of all. And here you go, the hip joint. This is the ball and socket type of joint we are discussing about. So we can say that the ladder runs from plane joints that only glide. That means the chart started with plane, that gradually transitions through hinge joint, then pivot joint that move one way, to condyloid and saddle that move two ways, to ball and socket that moves every way. And trap about the knee right now. The knee is classified as a hinge joint, moving mainly in flexion and extension, but it is not a pure hinge because it permits slight medial tibial rotation that you see here, that the tibia can rotate a bit on full extension, which is called the screw home mechanism, a little locking twist when you straighten your leg, and a cartilage distinction. The knee contains menisci, the C-shaped pads, not a complete articular disc. The temporomandibular joint, the TMJ, your jaw joint, and the sternoclavicular joint, the joint between clavicle and sternum, are the ones with true complete discs. Finally, know the knee's internal hardware. The intracapsular structures are the ACL, the PCL, and the medial and lateral menisci, while the collateral ligaments are extracapsular or capsular on the outside. So the knee is a hinge with a twist, cushioned by C-shaped menisci, not a full disc. Don't get confused about the names. I have just demonstrated it so that you can understand what extracapsular or intracapsular means. But IMAT won't ask about the specific names on the exam, so you can forget them, but you need to understand the concept, because sometimes IMAT can ask statement types, true-false questions, and for that you need to understand the underlying mechanism, because without that you won't be able to answer a question properly. So let me repeat. Knee is classified as hinge, but it permits medial rotation a bit. Then knee contains menisci, and TMJ, sternoclavicular joints, have true discs. The last one is knee has intracapsular structure and also extracapsular structure. If you can remember them. If you forget them, that's okay too. No problem with that. This second trap corrects a lazy assumption about synovial fluid. It is not simply plasma leaking into joint. It is actively made. Type B synoviocytes. Cells of the synovial membrane actively synthesize and secrete hyaluronic acid, and the molecule that makes the fluid thick and slippery. Meanwhile, type A synoviocytes are macrophage-like phagocytes that clear debris, the cleanup crew that eats stray particles. So there are two synovial cell types. Type B makes the lubricant, type A cleans the joint. That hyaluronate viscosity is what gives the fluid its lubricating power. And for reference values, the exam may test normal fluid in case of synovial fluid is pale yellow with fewer than 200 cells per cubic millimeter and no red cell. The number is not that important, but it doesn't contain red cell. You need to remember it. And the example talks about the shoulder joint. You know the shoulder joint? Let's say this is the shoulder, then you have humerus. So this joint we're talking about. This is also called glenohumeral joint. So the pearl capturing a deep principle regarding this. It captures the trade-off between mobility and stability. So the glenohumeral joint we were discussing has the greatest range of motion in the whole body. It's written here because the glenoid fossa, its socket. That means there will be a socket like this and there will be head of humerus getting attached to it. And The socket or the fossa is shallow relative to the large humeral head or the ball. A shallow socket lets the ball swing freely in every direction. But nature never gives something for nothing. That same shallowness makes it the most frequently dislocated major point, because a shallow socket holds the ball loosely. So the shoulder buys its freedom with fragility. Compare the hip, also the ball and socket type, but with a dip socket, far more stable and far harder to dislocate at the cost of less range. And one more comparison for completeness. Plain joints have the least movement among synovial joints, though fibrous sutures are less mobile still, being essentially immovable. So across all joints, mobility and stability sit on a seesaw. The freer the joint, the easier it dislocates. The shoulder is the extreme of freedom and therefore of instability as well. This is the payoff slide. Slide 60, where every mechanism we built becomes a disease you can reason through instead of memorize. The secret is that each disease is just the normal machinery running wrong, and the blood test, serum calcium, ALP are the fingerprints that tell them apart. Read the table disease by disease. First, osteoporosis. Core mechanism, resorption exceeds formation, giving normal mineralization but reduced bone mass. Blood, serum calcium normal, ALP normal, and the distinguishing feature is that post-menopausal estrogen loss is the commonest cause. Exactly the estrogen OPG RANKL chain we built 2 slides ago. So normal bone, just too little of it, and normal blood. Second comes rickets or osteomalacia. Actually, these are not interchangeably used. Rickets and osteomalacia, they are different quite a bit. But mechanism is same, and that is vitamin D deficiency. So osteoid is produced but not mineralized. Blood calcium low or low normal. ALP elevated. Feature soft bones with both legs in children and bone pain in adults. So the scene is in rickets and osteomalacia, the bones are soft, unhardened, calcium low, ALP high. Third comes primary hyperparathyroidism. Mechanism, autonomous PTH excess drives osteoclast resorption, a gland stuck on. Blood calcium high, ALP elevated. Feature, high calcium with low phosphate plus renal stones. So the bank is being emptied non-stop, so blood calcium soars. Fourth is Paget disease. Mechanism would be chaotic accelerated remodeling, producing disorganized mosaic bone. Breakers and builders both racing but out of sync. Blood calcium is normal, ALP very elevated, the sky-high ALP being the clue. Feature, enlarged, deformed, warm bone, commonly in the skull and pelvis. Fifth is renal osteodystrophy. Mechanism, kidney failure removes 1-alpha hydroxylase, causing secondary hyperparathyroidism. The activation organ failure we flagged on the vitamin D slide. Blood calcium would be low, ALP would be elevated. Feature, high phosphate with low calcitriol. So scan the calcium column like a fingerprint. Normal in osteoporosis and Paget disease. Low in rickets and renal disease. High in hyperparathyroidism. That one column already narrows almost every question. That means this calcium is the most important clue we need to remember. As we have seen the diagram beforehand, we're not going to use any charts or diagrams here. We're going to describe it with our prior knowledge. So let us walk the renal failure chain slowly because it ties 3 earlier slides into one story. Kidney failure leads to loss of 1-alpha hydroxylase activity. The kidney was the organ for activation of vitamin D. So failure kills that step. Therefore, calcidiol cannot convert to calcitriol, and active vitamin D falls. With no active vitamin D, intestinal calcium absorption falls, reducing hypocalcemia. Low blood calcium because active vitamin D was what let you absorb calcium from food. And this low serum calcium drives parathyroid stimulation, reducing secondary hyperparathyroidism. The parathyroids, sensing low calcium, ramp up PTH. That is written here. We call it secondary, written secondary here, because the gland is reacting to a problem elsewhere. The gland is not broken itself. Then persistent PTH excess drives osteoclast resorption, producing renal osteodystrophy, the bone disease of kidney failure. And in parallel, the failing kidneys cannot excrete phosphate. So serum phosphate rises. So the chain is that activation step. No active vitamin D. Poor calcium absorption, low calcium, reactive high PTH, bone breakdown plus rising phosphate. Every link is a mechanism you already own. The disease is just the chain assembled. The central trap of this slide. Students expect osteoporosis, the bone disease, to show abnormal calcium. It doesn't. In osteoporosis, the remaining bone is fully mineralized and calcium homeostasis is intact. So serum calcium is normal. The problem is the amount of bone, not the calcium balance. So the blood looks fine. Use that as a diagnostic fork. A low serum calcium points toward vitamin D deficiency or renal disease instead. And a high serum calcium points toward hyperparathyroidism or malignancy, not osteoporosis. So if a stem describes fragile bones but explicitly gives you a normal calcium and normal ALP, it is steering you to osteoporosis. If the calcium is abnormal, the answer is something else. Normal blood with weak bones equals osteoporosis. And a pearl that gives you a razor-sharp diagnostic fingerprint. Recall from the PTH slide that PTH raises serum calcium but causes phosphaturia, dumping phosphate into the urine. So serum phosphate falls. Therefore, high calcium with low phosphate is the fingerprint of primary hyperparathyroidism, the two minerals moving in opposite direction, calcium up and phosphate down, which is exactly what runaway PTH does. Now the discriminator. High calcium with high phosphate suggests a different cause, such as vitamin D toxicity, where both rise together because too much vitamin D pulls both calcium and phosphate in from the gut. So when a question gives you a high calcium, do not stop there. Look at the phosphate. High calcium with low phosphate means hyperparathyroidism. High calcium with high phosphate means think vitamin D excess instead. The phosphate direction splits the two apart. We finish where every good study session should finish, with a rapid pass over every key term, so the whole lecture consolidates into a single sweep. Watch the table and let each line trigger the full idea we built, because if you can expand each of these one-liners into its mechanism, you own this topic. Let me run them. The axial skeleton. 80 bones of the central body axis. Skull 22, spine 26, cage 25, hyoid 1, ossicles 6. The hyoid bone, the only bone articulating with no other bone. The diaphysis, the shaft with the compact wall around the medullary cavity. The periosteum, the outer sheet that covers all bone except articular surfaces and drives width, growth, and repair. The osteon, concentric lamellae around a Haversian canal, absent from spongy bone. Canaliculi, the channels connecting lacunae for osteocyte communication. The Volkmann canal, perpendicular and unringed, unlike Haversian canals. The osteoid, unmineralized matrix having type 1 collagen piling up in vitamin D deficiency. Hydroxyapatite, the mineral crystal giving compressive strength, while collagen gives tensile strength. The osteoblast, which is builder from mesenchyme, ALP marker carrying the PTH receptor. The osteocyte, the entrapped osteoblast, the mechanosensor, the most numerous. Osteoclast, multinucleated breaker from the monocyte lineage. Trap marker, ruffled border carrying the calcitonin receptor. Cathepsin K, the osteoclast protease that chews collagen after acid dissolves the mineral. RANKL and OPG, the osteoblast's accelerator and brake. That's why written RANKL activates, OPG blocks. And their ratio sets resorption rate. Intramembranous ossification. Bone straight from mesenchyme, the flat skull bones, mandible, and clavicle. Endochondral ossification. Bone replacing a hyaline cartilage model. All the long bones are formed in endochondral ossification process. The epiphyseal plate, the hyaline growth disc, and the zones are RPHCO. Growth occurs in proliferating zone. Woven bone, the immature first bone of development and fracture callus. The PTH, or parathyroid hormone, it raises calcium, acts on osteoblasts, not osteoclasts. Lower phosphate. Then comes calcitonin. It lowers calcium, directly inhibits osteoclast. Calcitriol, active vitamin D, made by the renal enzyme under PTH, enabling mineralization. Estrogen raises OPG, and its loss drives post-menopausal osteoporosis. Hyaline cartilage, type 2 collagen at articular surfaces and growth plate. You can find it. And the articular form lacking perichondrium. Fibrocartilage, type 1 and type 2. No perichondrium present. In the disc, menisci, pubic symphysis. Elastic cartilage, type 2 plus elastin in the ear and epiglottis, always with perichondrium. The gomphosis, the tooth in socket joint, the body's only example. The synchondrosis, a hyaline cartilage joint like the growth plate, immovable and usually temporary. Symphysis, a fibrocartilage joint like the pubic symphysis and the discs slightly movable. The synovial joint, defined by its fluid-filled cavity. All diarthrosis. Synovial fluid, hyaluronate-rich, actively secreted by type B synoviocytes. The meniscus, the C-shaped knee pad, distinct from a complete disc. Ball and socket joint, multi-axial, shoulder and hip is an example, and shoulder having the greatest range of motion. Osteoporosis, where bone mass is reduced, mineralization even stays normal after that, and normal serum calcium. And osteomalacia and osteoporosis are distinguished by this. And finally, osteomalacia and rickets. It has defective mineralization problem. The calcium level is low normal, ALP is elevated, and the bones become soft. That is the entire lecture in one sweep. If every one of those terms just unfolded into its full mechanism in your mind, you're ready for skeletal questions the IMAT can build. If any of them stayed flat, go back to that slide and rebuild it. Because a term you can only recite is a term that exam can trap, while a term you can explain is a term you own. This has been IMAT Tutor. Master the mechanism and marks will follow. We have built the whole machine. Now we prove it works by solving 10 exam-style questions the way you must solve them under pressure. For each one, I will read the stem, then I want you to pause the video and commit to an answer before I speak. Because a question you have actually attempted teaches 10 times more than one you passively watch. Then we reason it out together, walk every option, and eliminate the traps. This is where the marks are actually earned. Question 1 reads, which of the following bone cells is derived from monocyte macrophage hematopoietic lineage? The options are osteoblast, osteocyte, osteoclast, osteoprogenitor cell, and chondroblast. Pause and choose. This question is a pure test of the family tree fact we hammered on the bone cell slide, and it is a favorite because it looks like simple recall, but it is really a trap for anyone who assumes all bone cells share one origin. The key word here is lineage, the cell's ancestry. Remember our grade split. 3 out of the 4 bone cells come from mesenchyme, the connective tissue family, and only 1 comes from the blood family. Walk the options. Option A, osteoblast. The builder is mesenchymal. Let's write M for it. Option B, the osteocyte is just a retired osteoblast walked into the matrix, so it is also mesenchymal. Then option D, the osteoprogenitor cell. This is the apprentice builder. This is also mesenchymal. And option E, the chondroblast, the cartilage maker, also springs from mesenchyme. That leaves option C, the osteoclast, the demolition crew, which alone comes from the hematopoietic monocyte macrophage lineage, the same family as your immune macrophages, because osteoclasts form when monocytes fuse. So the answer is C, the osteoclast. The whole question turns on one fact. The breaker is the odd one out, born from blood, while every builder type cell is born from mesenchyme. If you knew that split, this was a 5-second question. If you did not, all 5 options looked equally plausible, which is exactly the trap. Question 2 reads, a 70-year-old woman presents with severe back pain. Imaging reveals multiple vertebral compression fractures. Her serum calcium is normal and bone biopsy shows reduced total bone mass with normal mineralization of remaining bone. Which condition best fits this presentation? The options are rickets, osteomalacia, osteoporosis, hyperparathyroidism, and Paget's disease. Pause and choose. This is a classic clinical vignette, and the exam has buried the answer in the details. So read them like clues. Clue 1. A 70-year-old woman. That means post-menopausal. Let's write it down. The exact group with estrogen loss bone disease. Clue 2. Vertebral compression fracture. That means bones are fragile. Clue 3, and this is the decider. Serum calcium is normal. Clue 4, biopsy shows reduced bone mass, but normal mineralization of what remains. Now reason. Rickets, option A, is a childhood disease of soft, unmineralized bone. Wrong age and wrong biopsy. So, no. Osteomalacia, option B, is the adult version. Defective mineralization, but the biopsy explicitly says mineralization is normal. So B is eliminated by that one word. Hyperparathyroidism, option D, would show high serum calcium, but ours is normal. So, no. Paget's disease, option E, shows chaotic bone with sky-high ALP, not simply reduced mass. So, no. That leaves option C, osteoporosis. Reduced quantity of normally mineralized bone with normal calcium in a post-menopausal woman, and every clue fits here. So, our final answer would be option C, osteoporosis. The single most powerful clue was the normal serum calcium with normal mineralization, which is the fingerprint that separates osteoporosis, a quantity problem, from osteomalacia, a quality problem. Notice how the exam put osteomalacia right next to it as a bait. The blood test and the biopsy wording are what split them. Question 3 reads, during endochondral ossification, what is the correct sequence of events in the epiphyseal plate zones from epiphysis toward diaphysis? Then 5 options are given with mentioning zones in different order. So pause and choose. This is a pure sequence question and it rewards the chant we built. The chant was RPHCO, from the epiphysis toward the diaphysis. That means from the end toward the shaft. R, that means reserve first, you see in the image, because the resting cartilage anchors the blade to the bone end. Then proliferating, where the cells divide and stack, the growth engine. Then hypertrophic, where they swell. Then calcified, where the matrix hardens. Then ossification, where bone finally replaces cartilage. Now scan the options for that exact order. Option A starts with proliferating. Wrong. Resorb must be first. So option A is not correct. Option C starts with hypertrophic. Wrong. Option D starts with calcified and even puts reserve fourth. Badly scrambled. Wrong. Option E is subtle trap. It starts correctly with reserve but then swaps the next two, giving hypertrophic before proliferating, which is backwards. So E is out. Only option B gives the clean RPHO. So the answer is B. Notice how option E was designed to catch someone who half remembered the chant, getting reserve right but fumbling the middle. That is why you memorize it as one locked 5-letter unit, RPHCO, not as loose words you reassemble under stress. The chant defeats the scramble trap. Question 4 reads, which of the following is the only synovial joint component that is unique to synovial joints, that is not present in any other joint type? The options are articular cartilage, ligaments, joint cavity, periosteum, fibrous capsule. Pause and choose. The key word is unique, meaning found in synovial joints and nowhere else. So we test each option by asking, does this appear in any other joint type? To get a clear picture, let's bring the diagram as well. It will help you answer it more easily. Option A, articular cartilage. No, because cartilaginous joints like synchondrosis are made of cartilage too. So cartilage is not unique. Option B, ligaments. No. Fibrous joints such as syndesmosis are built from ligaments, so ligaments appear elsewhere. Option D, periosteum. No, the periosteum covers essentially all bones regardless of what joint they form, so it is everywhere. Option E, fibrous capsule. No. A fibrous outer layer shows up in other joint arrangements too. That leaves option C, the joint cavity, that you also see in here, the joint cavity. The fluid filled space lined by synovial membrane, and this truly exists in no other joint type because fibrous and cartilaginous joints glue the bones directly together with no gap at all. The cavity is the defining feature. This is a definition question in disguise. The one thing that makes a synovial joint synovial is the fluid-filled cavity. Every other option is a component that synovial joints happen to have, but other joints have them too. Unique means the cavity, always. Question 5 reads, parathyroid hormone acts on bone to raise serum calcium. Which cell type does PTH directly bind in bone to achieve this effect? Options are osteoclast via RANK receptor, osteocyte via PTH1R receptor, osteoblast via PTH1R receptor, osteoprogenitor cell via calcitonin receptor, chondrocyte via PTH1R receptor. Pause and choose. This question directly tests the trap we drilled hardest. Remember, PTH acts on the builder, not the breaker. The word directly is doing all the work here. Option A is the seductive wrong answer. The osteoclast via RANK, because it feels right that the bone resorbing hormone hits the bone resorbing cell, but osteoclasts have no PTH receptor. They are activated indirectly. So A is the trap and it is wrong. Option D pairs the osteoprogenitor with a calcitonin receptor, which is simply mismatched. Wrong. Option E puts the receptor on a chondrocyte, a cartilage cell irrelevant to this pathway. Wrong. That leaves B and C, both naming the correct PTH1R receptor, but on different cells. Option B says osteocyte. It is true that osteocytes carry some PTH1R, but the primary intended mechanism of PTH-driven resorption is the osteoblast raising RANKL to activate osteoclast. So the best answer is option C, osteoclast via PTH1R receptor. 2 lessons from this question. First, the classic trap in option A, PTH on osteoclast, is wrong because the pathway is indirect through the osteoblast. Second, when 2 options are both partly true, as B and C were, pick the one describing the primary intended mechanism, which is osteoblast signaling to osteoclast via RANKL. Let me show you a reminding diagram so that you can remember it permanently. You see in the image, osteoblast expresses RANKL. The RANKL binds to RANK An osteoclast precursor becomes osteoclast and it starts demolishing the bone. And the PTH will work on the PTH1R receptor found on the surface of osteoblast. Question 6 reads, the pubic symphysis is classified as which type of joint? Options are fibrous joint and suture, cartilaginous joint, synchondrosis, cartilaginous joint, symphysis, synovial joint, condyloid, fibrous joint, syndesmosis. Pause and choose. The name itself is the biggest clue. Pubic symphysis contains the word symphysis, which is a specific joint type, and that is almost certainly not an accident. But let us confirm by reasoning, not just word matching. A symphysis is a secondary cartilaginous joint made of fibrocartilage that you see here. This is the pubic symphysis, and which is slightly movable. Test the options. Option A, fibrous joint suture. But fibrous suture is skull only. Wrong location and wrong tissue. Option E, fibrous syndesmosis is the tibia fibular or radioulnar type. Wrong. Option D, synovial condyloid would need a joint cavity, but the pubic symphysis has none. It is a solid fibrocartilage pad, so wrong. Now the tricky pair, B versus C. Both cartilaginous. Option B, synchondrosis uses hyaline cartilage and is immovable like the growth plate, but the pubic symphysis uses fibrocartilage and moves slightly, so it is not synchondrosis. Option C, symphysis is exactly fibrocartilage, slightly movable. That is our joint. And remember why it moves at all. During childbirth, relaxin softens it to widen the birth canal. That means this one. The clearest proof that a symphysis is amphiarthrotic, so slightly movable, not fixed. The trap was option B, synchondrosis, for anyone who lumped all cartilage joints together. Hyaline and immovable means synchondrosis, fibrocartilage and slightly movable means symphysis. Question 7 reads, which type of cartilage covers the articular surfaces of bones in a synovial joint? Options are elastic cartilage, fibrocartilage, hyaline cartilage, calcified cartilage, and yellow cartilage. Pause and choose first. Then let me explain. This is one of the most tested single facts in the whole topic, and it has a built-in trap. The articular surface, the actual cap on the bone ends, is hyaline cartilage. You can see this image. This is the articular cartilage. Now walk the options. Option A, elastic cartilage, is for the ear and epiglottis, things that bend and spring back, not joint surfaces. Wrong. Option D, calcified cartilage. It is a temporary scaffold in ossification, not a joint surface. Wrong. Option E, yellow cartilage, is just another name for elastic cartilage, so it is a disguised repeat of A. Wrong. Now the real trap, option B, fibrocartilage. Students pick it because they correctly remember the knee menisci are fibrocartilage, and they wrongly conclude the joint surface must be fibrocartilage too. But the menisci are extra pads wedged inside the joint, kind of like this. The surface itself, the cap on the femur and tibia, it is hyaline. So option C, hyaline cartilage, is correct. The exam deliberately baits you with fibrocartilage because you know menisci exist. Hold the distinction firmly. Menisci are intra-articular cushions, but the articular surface that actually caps the bone is always hyaline. Surface equals hyaline, pads equals fibrocartilage. Question 8 reads, a patient with chronic renal failure is found to have elevated PTH levels, low serum calcium and elevated serum phosphate. Which mechanism best explains the low serum calcium? The options are given below. Pause and choose. This is exactly the renal failure chain we walked on the pathology slide. The kidney is the activation site for vitamin D. To get a clear perspective, let's bring an image. Here you go. As we were saying, kidney activates vitamin D using enzyme 1-alpha hydroxylase. If the kidney fails, the activation step dies. Now test the options as explanation for the low calcium. Option A, increased osteoclast resorption would actually raise calcium, not lower it. so it contradicts the finding. Wrong. Option C, decreased PTH contradicts the stem itself, which says PTH is elevated, so it's wrong too. Option D, increased calcitonin would lower calcium, but it's not the mechanism in renal failure, and nothing here drives calcitonin. Wrong. Option E, increased phosphate absorption does not explain low calcium and gets the phosphate mechanism wrong anyway. That leaves option B. The failing kidney cannot convert 25 hydroxyvitamin D calcidiol into 1,2,5 dihydroxyvitamin D calcitriol. So intestinal calcium absorption collapses and blood calcium falls. And that low calcium is exactly what then drives the elevated PTH, the secondary hyperparathyroidism. The trick to multi-step questions like this is to find the root cause, not a downstream effect. The elevated PTH is a consequence of low calcium, not any other reason. And the low calcium is a consequence of the dead activation enzyme. The root is the kidney failing to make active vitamin D. Trace the chain to its source and the distractors fall away. Question 9 reads, which of the following statements about the structural unit of compact bone is correct? And 5 options are given. First of all, pause and choose one answer from the options. This is a find the true statement type question. So we test each one against the osteon anatomy we drew. Let's bring the osteon diagram as an aid. Option A says the osteon consists of concentric lamellae surrounding a Volkmann's canal. But osteons surround the Haversian or central canal. Volkmann canal run perpendicular. Like if this is a Haversian canal, then Volkmann canal will connect two Haversian canals perpendicularly. So A is false due to that reason. Option B says spongy bone contains osteons, but we have said it over and over. Spongy bone has no osteon, only trabeculae. So B is false. Option D says interstitial lamellae are the most recently formed bone, but they are the opposite. They are actually the remnants of old resorbed osteons. So D is false. Option E says the Haversian canal contains only nervous tissue, but it carries blood vessels and nerves both. So only is wrong, making E false. You see in the center, there are blood vessels and nerves together. And that leaves us with option C. Canaliculi or canaliculi connect adjacent lacunae to allow osteocyte communication. So these are the canaliculi, which is exactly right. Those hair-thin tunnels linking the cell caves so buried osteocytes can feed the signal. C is the correct statement. Find the true statement questions are really 4 traps and 1 true. Each wrong option here embeds a classic error we already dismantled. The Volkmann mix-up, the spongy bone osteon myth, the interstitial lamellae age error, and the Haversian contents trap. Because you build the anatomy properly, you could shoot down all 4 and be left with the truth. Question 10 reads, a 12-year-old child sustains a fracture through the epiphyseal plate of the distal radius. What is the most significant concern regarding this injury? Pause for a while and try to answer it yourself. Alright, now 2 details in the stem are the whole question. A 12-year-old, meaning still growing, and a fracture through the epiphyseal plate. That means the growth plate itself. So the exam is asking what is special about breaking a growth plate in a child. Let's bring the image to support our explanation. The growth plate is the hyaline cartilage engine of longitudinal growth. You see here. Specifically, it's proliferating zone. Now weigh the option. Option A, B, and E. Articular cartilage damage, hemarthrosis, and joint instability are all possible co-injuries, but none is specific to the growth plate. They could happen with many wrist injuries, so they are distractors. Option D, loss of red marrow function is not the concern. That leaves option C, disruption of longitudinal bone growth, which is exactly the unique danger. damage the proliferating chondrocytes, and that bone may stop lengthening, giving a limb length discrepancy or an angular deformity as the child grows. You can see here. And this is Salter-Harris fracture. And the reason pediatric growth plate fractures are treated so urgently is precisely this risk of arrested growth. The exam skill here is to notice the two loaded words, child and growth plate, and ask what is unique about their combination. The other options are real injuries but generic. Only C is specific to breaking the growth engine. All right, with that, finally we come to the conclusion of our video. 10 questions, 10 mechanisms proven under exam conditions. Notice the pattern across all of them. The IMAT rarely tests a bare fact. It tests whether you can eliminate a well-designed distractor, and every distractor was one of the exact traps we named in the lecture. That is why we build the mechanism so carefully, because a mechanism you understand lets you shoot down 4 wrong options and walk to the right one with confidence. Go back and reattempt any question you missed, rebuild the slide behind it, and it will not catch you twice. This has been IMAT Tutor. If you like the video, please drop a like, do subscribe, and if you have any question regarding IMAT, feel free to ask them in the comments. And also, our IMAT complete package is ready, and you can check the link in the description. Thank you.